Wonderful. All righty. Brian, if you're good to go, I think we can go ahead and start with Charlie.
Absolutely. Everything's all set on my end. Welcome, everyone. Welcome.
Okay. Well, hello, everyone, and welcome to this worldwide conference as it draws together so many talents across the state and across this nation, perhaps, for those who are striving to build and strengthen the agriculture community. My name is Charlie Hunsicker. I'm the Director of Parks and Natural Resources here with Manatee County Government. As you can tell by the gray hair, I've been involved with Manatee County for about 41 years. And in that role, we've been able to touch so many things relative to environmental quality, water quality, and promotion of things that drive our economy, not only from a tourism level, but from an agriculture level. Cortez is one of our last working waterfronts of Manatee County in the state of Florida. And by that, we work as a through the Board of County Commissioners to support that industry and to make whatever effort we can to promote and remove the barriers to productive work in the agriculture industry. I wanted to, in part of welcoming you, outline some of the things that the Board of County Commissioners are doing to work their side of the political spectrum to invoke and encourage investments in agriculture. One such move this year was to secure a policy statement from the Florida Association of Counties, a group of all 67 counties that work in concert on joint legislative platforms with the Florida legislature. And with that, they also carry those policy statements at the federal level when it's appropriate. There are several committees, obviously, to deal with the many issues of Florida, but one of those committees is the Agriculture and Rural Affairs Committee that does address agricultural policies for all manner of livestock, growers, producers, and in a small niche, in a way, aquaculture is one of those producers. And Manatee County government, myself and commissioners worked through last year to obtain support from the broader Association of Counties for a policy position on aquaculture. And with the extensive help of Angela Collins and her artful crafting of finding the divide between scientific terminology and layman's terms as our area Sea Grant extension agent. She assisted me with developing a series of policy statements that I just would kind of like to repeat here as an opening statement for your conference coming up. Because as I look at the presentations for nutrient removal, water filtration, seagrass enhancement, nutrient credits and values, and carbon credits, all of those things function and revolve around the research needs to establish those parameters. Well, the Florida Association of Counties this year agreed to adopt a policy statement on this, covering five major areas. I've asked Dinorah to make those available to each of you throughout this conference and at the end as an attachment, but I'll just summarize them very quickly. As a policy statement to carry to this legislature and legislators in the future in Florida, we have asked a review of submerged land leasing requirements and revisions of restricted and outdated policies for leaseholders in our area waters. With so much water available and so few leases out there, what are the regulatory obstacles there? We've also asked that the Florida Department of Agriculture and Consumer Services, or the Farm Service Agency, evaluate the establishment of viable crop insurance program for aquaculture. I know that a crop insurance program was tried some years ago and for several reasons broke down in terms of reliability and dependability. And we believe that there still is a viable opportunity for crop insurance to cover losses due to the inability to harvest during red tide or other harmful algae bloom moratoriums. We also believe that there are other times when hurricanes will come through and really impact a crop, scattered across the bottom. And yet there's no viable agricultural insurance program for that. And we're working constantly and consistently to make that happen. We also want to direct the Florida Department of Environmental Protection to consider the use of live clams, I'll read from this, to enhance the success of seagrass impact mitigation requirements. That's going to take a little leap between what we know anecdotally and what we believe the regulators can incorporate. For of course, when we're dealing with seagrass mitigation and its high costs, if we could find the linkage between clams and seagrass mitigation success, we could adjust the mitigation requirements and thus buy down the cost of seagrass mitigation when clams are used in conjunction with traditional seeding and planting methods. We'd also like to encourage FDEP, FWC, and the relevant state agencies here to promulgate the standards for the deployment of bivalve shellfish for large-scale water quality improvement and nearshore habitat creation. Now, it's become quite a popular move among all of us laymen to say, well, why don't we seed tens of thousands or hundreds of thousands of clams, live clams in area waters, and oysters too, to see the recovery in the nearshore and shallow water environments for water quality purposes, letting Mother Nature take all those natural efforts. So I may just be going over all the talks that you're going to be doing through the session, but I want to say that it was important for us as a county to ensure that all of Florida recognize the value of aquaculture, not here just in the southeast or southwest, but along the Atlantic coast, up in the Panhandle. Of course, there's a massive effort underway to try to reestablish the viable oyster industry that was there through use of funding through the BP oil spill and proceeds and other grants available. And we're fully supportive of that. I think it was time that the Florida Association of Counties woke up to the viable industry that's out there, even though it's relatively a niche market. But still, up until this point, when this was passed on December 2nd of 2020, there was no policy statement on aquaculture, none. And so I think we've done a great service, I think, to move in that direction. Our Board of County Commissioners here are also enthused to work at the federal level. Last year, Angela and I and Dr. Aaron Welch went to Washington, D.C., or was it 2 years ago now? I can't remember. But we went to Washington, D.C., and we met with individuals with the USDA and I think it was FDA, to address the type of sampling methodologies and accelerated methodologies to detect presence and absence of harmful concentrations of harmful algae in oysters, and to go beyond the so-called mouse method, which was a very archaic and frustrating and lengthy time. They moved in that direction. I think we had something to do with that. We have been working with our congressional delegation, including the House and Senate, to move and support and identify the importance of aquaculture. And we're seeing that come to fruition in some of the federal legislation that's coming through Senator Rubio, and also sponsored here with Congressman Buchanan and Congressman Mast over on the East Coast. Of course, our partners in crime, in government over in the Indian River Lagoon are also working very hard to find these natural means to complement water quality treatment and water quality control. So it's a big mouthful, right? But from our standpoint, I'm pleased, honored to work with the Extension Program and with our area scientists as a go-between with our Board of County Commissioners, who are just actually meeting today to talk about our capital improvement programs across hundreds of millions of dollars. And there are times, not often, but there are times when aquaculture and seafood and stuff like that is kind of like, it's over here, this direction, because we are focused on transportation, we're focused on education and health programs and all those things. But at the same time, we recognize the value of aquaculture. We recognize the value of heritage, of the heritage of the industry, the people who came before us, and the farmers who can thrive ahead of us to make it the best setting to make that industry, our industry, successful. So we're there. I want to make sure that I give a shout out to Angela again for her tremendous efforts and her successor, John Steveley, who set the tone for why aquaculture is important It's here to stay, and we want to work to make sure that it stays forever, quite possible in our area. So thanks again for my ability to welcome you to the conference. And if you haven't traveled to Manatee County, I'm sure you have, but we've got plenty of things going on, not only out in the water, but on the land as well. I invite you to visit any one of our preserves, some of our parks. You know, that's kind of my thing. And welcome me to enjoy many of those areas here across Southwest Florida and in Manatee County in particular. So thank you very much, and have a great conference. I've got to go that way. Okay.
Thank you so much, Charlie. That was a wonderful welcome message and so much information, important information to have. So we really appreciate having you on board and working with you for sure.
Thank you. Thank you again. So bye.
Bye-bye. All righty. Brian, so now I just need to share my screen. All righty.
Should be all set.
Perfect. Let me go ahead. Let me try this first and see. Quickly check with you, Brian, that this looks well, like my slides are all in there.
Yep, looks good.
Perfect. All righty, welcome everyone. Good morning. We're super excited to kick off this meeting this morning. We have some really interesting presentations on some very relevant topics over the next couple of days. And despite our attempts to have this workshop last year in person, well, you know the situation. So we rather have everybody safe from their homes. And yeah, thank you so much for joining us. We know it's not the same as being in person, but we're still going to make the best out of this. My name is Dinorah Chacin. I am going to be the moderator of this workshop during the next couple of days. You'll be hearing and listening a lot of me. If you have any questions, please feel free to message me directly. I am serving as the acting executive director of the Gulf Shellfish Institute, GSI. And if you're not super familiar with GSI, it is a Florida not-for-profit corporation. Our mission is to facilitate, support, and encourage the increased production of shellfish in Florida and the Gulf region in order to boost economic and environmental benefits. We do this through cooperative industry-driven research and outreach, and we do this by partnering with lots of different groups here in Florida and in the region as well. So why are we here? So we've heard from so many different sources, or we can also speculate from other Bible species that clams can perform a variety of ecological services in the ecosystem. Some of the ones that are most frequently heard is that they clean our waters because they can filter feed. Clams can remove nitrogen from coastal waters. Maybe they can sequester also carbon from the atmosphere, and many other benefits that I haven't even started to mention. But all these different ecological services are often the reason why there's so much interest in restoring populations of these clams. Now, our experience with clam restoration has led us to realize that in spite of many claims of the ecological services these wonderful clams perform, there's still a lot that we do not know, but we need to know in order to be able to quantify the services. So the overall goal of this workshop is really to examine each of these ecological services, document what is really known about them regarding mercenary species, and determine what researches still remain to do to fill in these knowledge gaps. As we all know, clam restoration is becoming super popular, especially here in Florida. And really filling in these research gaps is what is going to allow us with certainty to say clams are performing XYZ ecological services. So the overall goal is to really gather all minds together over the next couple of days and talk about this topic, identify those needs, and especially for restoration efforts, so we can support those efforts. In doing this, we're hoping to open a line of communication among the different groups involved, whether you work with specific restoration group, a nonprofit, an agency, your farming industry member, scientists, regulatory. We're just hoping that this is going to allow the conversation among all of us. And this will likely not be the first nor the last workshop talking about this topic. We know there's one coming up on the East Coast. So we're hoping that this is just one step forward, bridging that communication among the different groups involved in restoration of clams. So that way, when we're really talking about clam restoration, we're producing a coherent framework. We're all on the same page of what is it that we're really talking about, and what do we know and what we still don't know. And of course, we hope to summarize our findings from the next couple of days into a document that we can share with all of you guys, and everybody can benefit from the discussion. Now, to start getting your minds thinking, I know it's very early, but we sent out a pre-survey. We're super excited about this workshop. We have about 121 participants. This was far exceeding our expectations. I almost feel that doing it virtually, we have been able to reach out so many more people than if we were in person. Of course, we don't have all the responses. We have only like about 32 responses so far, but I just wanted to give you a bit of an insight of what we have found out so far from who is present. We have a lot of interest from academic researchers, government regulatory, nonprofits. I hope that in the audience, there's also more representative from the industry and farming. We ask you about your agreement with the following statements. Do you understand clam biology and ecosystem services? Most of the responses were on the agree spectrum. Also, do you understand the goals behind ongoing restoration efforts? Most of the responses were I agree. Are you supportive or, of the restoration activities currently and future? Most of the responses were on the agree aspect. We ask you how much you agree with the statement that states hard clam restoration activities can improve water quality. We definitely strongly agree there. Benefit the shellfish aquaculture industry. We also agree. Produce benefits supported by existing research. We saw some of the agree, also some neither agree or disagree, and require additional research to quantify these benefits. Strongly agree. So this just to give you an idea of what we're thinking. We know that hard clam can probably have really good impacts in our environment, but we definitely need more research to really understand what's happening. And lastly, we ask you about ecosystem services. Which one do you think are enhanced by these projects? And water filtration was the most popular by far, but definitely a lot of the other topics that we're going to be covering in this workshop are also in the agree. And there's also wanted to point out to your attention that there was a lot of I need more info to decide, which I think is great because we really do need more information. Then I ask you guys to list 3 research priorities you think should be addressed to maximize the success of clam restoration efforts. And this is a word cloud. And so basically from all the written responses, it picks up and makes bigger like the word that it was mainly used. And I just want to bring to your attention that, of course, we see lots of clam, but also water filtration. So quantification of water filtration, nutrient removal. This has been widely cited as a need in order to support the current efforts. How do clams affect nutrient cycling? Are the efforts being effective? How are we measuring this? What are the metrics used to follow the tracking the stock clams? And what are the effects on seagrass as well and harmful algal blooms? So that was on point and very interesting. And we also ask you about your your top 3 concerns about ongoing and future clam restoration activities. And one of the words that popped the most, aside obviously from restoration and clams, was the lack. lack of funding to answer research priorities, lack of Florida-based data, lack of science on genetic diversity, larvae to responses of water quality, lack of a baseline, lack of long-term monitoring on project success. So with that, I just want to plant that seed in your mind as we continue discussing these topics of how much we still need to find out, and hopefully get your mind thinking about some good things that we can continue discussing today. I want to go ahead and thank our partners, Manatee County, the Restore Funding, Port Manatee, Florida Sea Grant, Tampa Bay Asteroid Program, Sarasota Baywatch, for all their support and also the funding to make this workshop possible. I just want to go over a few housekeeping items, and I'm sure most of all are familiar with Zoom by now or any other virtual platform. But we're working with virtual producers. So among the participants, you can find someone that's called virtual producer. So who are monitoring our meeting and making sure that things are working well. So if you encounter you're having any tech issues, feel free to message them or message me, and we will make sure we'll figure it out. Our representative is Brian Barber. Let's see, please remember to maintain yourselves on mute during presentations. There's a lot of us, and as much as I want to get everybody to talk, it could be distracting when somebody is giving a presentation to hear somebody else talking in the background. We're gonna have the chat box active, and right now there's a small introductory message saying, just welcome. You can state your name, your affiliation, and if you have any favorite clam-based dish, go ahead and do that. That's an icebreaker. This chat box is going to be active throughout the meeting. I know it could be distracting, so you're welcome to, just close it and just see it at the end of the presentation. But the idea is that if you have any interesting thoughts, comments throughout a presentation you want to say, feel free to go ahead and write it down there. Also, if you have questions for the speakers, please type it there. I'll try to keep track of them. And at the end of the Q&A, I will go ahead and go over each of the questions to make it easier for purposes of time and respect for everyone. Also, the chat box allows you to chat privately if you wish to. And let me see. Yeah, and I think like if we keep the chat box there, if we don't get to all the questions after a speaker because we've run out of time, somebody else from the audience may have an answer for you, or even the speaker can also take a look later and try to answer your questions. So we'll have a record of that. So with that, I'm going to conclude my introductory message, and I'm really happy that everybody is here. We're going to start with our first speaker, who is Dr. William Arnold. Bill is a native Floridian. He moved to St. Petersburg in 1985 and founded and led the Molluscan Fisheries Research Program at the Florida Fish and Wildlife Conservation Commission Fish and Wildlife Research Institute until 2009, all of this while obtaining his PhD at the University of South Florida College of Marine Science. During that time, he focused considerable research efforts on the distribution, ecology, and restoration of hard clams throughout Florida, with an emphasis on the Indian River Lagoon. He performed studies on the genetics, ecology, and larval dynamics of Mercenera mercenaria, Mercenera campechiensis, and their natural occurring hybrids. And his work has contributed substantially to our understanding of the history, environmental influences, and potential restoration of clam populations here in Florida. Bill then completed his career in fisheries science management as a Caribbean branch chief for NOAA's Southeast Regional Office, and he is now in private business. But Bill expresses love for these clams still, and he is very honored to contribute in any way possible to our understanding and management of these economically and ecologically important organisms. Bill, we're super lucky to have you as one of our speakers today. Please everyone help me welcome Dr. William Arnold to illuminate us on the history and goals of clam restoration. I will stop sharing my screen now. And Bill, you should be able to go ahead and share yours.
Thank you, Dinorah. I appreciate it. Yeah, so I'll share my screen. I admit It's been a while since I've given a scientific talk, so I'm a little nervous, but that's okay. Let me make sure I do this right. I hope you can see that. Can you see my title slide?
Not yet. Perhaps you have to click on that other, if you have multiple screens, there you go. Let's see. There, yeah, we can see it now. Perfect.
Yay for me. Okay, I did something right. Yeah, I'm going to talk about the very long-term history of clams in Florida, literally 10,000 plus years, because the Native Americans, far before the Europeans showed up, were utilizing clams and bivalves in general very heavily in their lifestyle. And what they have to contribute and what the history of clams in a variety of different ways have to contribute is really important to what everybody's trying to do, not maybe so much with pure aquaculture, but with aquaculture-based restoration and with restoration, and that's not really my favorite term, and I'll talk about that in general. So, as I say, and I'll repeat this, I probably will repeat myself a lot, but that's a good way to get a point across. The idea behind science is to build on what comes before. So you really need to know what your foundation is and build on that foundation. We don't have the money to be ignoring and repeating. So that's a large part of my motivation for giving this talk. So that's the title. As Dinorah said, I'm with the Gulf Shellfish Institute now. I'm on the board of directors, but I'm also in private business. So here's an overview of the talk. I always like to give a little outline so everybody knows what's coming. So when they're really, really bored, they'll know how close I am to being done. First is, as I mentioned, I'm going to talk about the pre-European, and that's mostly information, well, it's exclusively information we've obtained from shell middens. Then I'll talk about the hard clam fishery of Southwest Florida. For those of you who are not familiar with it, you'll be amazed, I hope, because it's pretty impressive. And then some early research and transplanting activities in Florida that had an incredibly important influence on things to follow. Then I'll talk about the Indian River hard clam fishery. I grew up on the Indian River Lagoon. Most of my work, not all of it, but a lot of the emphasis here is going to be on my clam work in the Indian River Lagoon. That's 30 years worth. A lot of time underwater, hundreds and hundreds of hours underwater, and thousands of hours on top of the water doing the work over there. So me and Dan Morelli in particular, but the other members of the FWI team, we put, and Mark Berrigan and John Gunter, we put a lot of time in over there and enjoyed it greatly. We really did. It was very challenging. As Dinorah said, I love clams. They're my favorite bivalve. They may be my second favorite animal next to my pet doggies. And they just have a lot to teach us. There are so many things we can learn that are contained within the bodies and shells of clams that we have so much to learn from and so much potential. Now, we talk about clams, we talk about clam restoration. A lot of the times what we mean is hard clams, Mercenaria, but there are a lot of clams out there, and that's also a really important thing to keep in mind, and I'm going to talk about that. So within the IRL, I'll talk about the development of an expansion of the wild fishery, and then its collapse and the causes of that collapse. And then I'll talk about development of hard clam aquaculture in Florida, which kind of went hand in hand. It's interesting what the potential relationships between aquaculture development and wild fishery development may be. Some of it's speculative, and some of this talk will be speculative, and other is very, very data-based. So that, just like the wild fishery, and this is in the Indian River Lagoon, its expansion and collapse, and then the redistribution to the rest of Florida or other areas in Florida. And then enhancement research, and I use the term enhancement, not restoration, and I'll talk about that too. And we put quite a bit of time and effort into this, and even some money. It was a severely underfunded effort, and that cost us some, but I do think we laid some nice groundwork that can be built upon. And then finally, considerations for future enhancement of hard, but not necessarily hard clam populations. And I really want to stress the importance of setting goals. And I'm going to talk about those goals relative to the talks that will follow. And I'm hoping that this talk will set up the following talks. And then a little bit on the population enhancement within the larger context. So that's the basic overview. So let's just dive right in. And like I said, I'm going to start with the pre-European era. These are shell middens. Some of them are just phenomenally well developed and expansive. You can see, you know, 15, 20, they probably aren't even at the bottom of it. And they're all over Florida and they're all over the southeastern United States. And they contain a lot of shells from a lot of organisms. Oysters and hard clams may be the predominant organisms. And of course, that's going to depend upon where you are. But there are a lot of, to say nothing of the vertebrates, fish, et cetera, fish bones, et cetera. There's a lot of bivalves and gastropods in there and other organisms. And, you know, Doug Jones and Irv Quitmeyer, I'll refer to them and their team at the Florida Museum of Natural History, Fred Andrus and a variety of people and in other areas have done a lot of work and extracted amazing amounts of information from these shell middens. The middens date back throughout coastal Florida, where I'll focus, at least 10,000 years and probably longer. So that's the part about the history being 10,000 plus years. And in the Indian River Lagoon, that's no exception. There are middens all over the Indian River Lagoon, all the way from Mosquito Lagoon down to Stewart. Not so much on the Banana River side, but certainly along the Indian River Lagoon. And in particular, Dan Morelli and I went with Doug and Irv to Honeymoon Hill on Merritt Island, which used to be just the biggest mountain, second biggest mountain on the Indian River Lagoon. All it is a midden, and it's a good 30, 40 feet high. Now it's covered with extremely expensive homes, but back then there was nothing on it. And we dug there, and what was really important and fascinating and needs to be kept in mind in your restoration enhancement efforts is the alternating nature of clams and oysters in that midden, which we believe reflects the alternating nature of the availability of clams and oysters in the Indian River Lagoon. So, you know, this is not a stable system by any means, and these bivalves in these middens reflect that. So it's really important to understand this long-term variability because it continues, it appears to continue to operate today. You know, and when I say today, the IRL has changed a lot. And I don't want to emphasize the IRL too much, but like I said, that's where I did most of my work. And in the last 10 years, maybe 15, it has really degraded. When, you know, my work there ended about 2009, 2010, when I moved out of FWI and went over to the National Marine Fishery Service. So the fact that things have changed there, folks work in the IRL probably need to do some additional work to see how well or how much the current situation reflects what we saw when we were working out there, because it could be drastically changed. And I also think it's important to know that based upon work by Irv and Doug, pre-European occupants in the southeastern United States overexploited these bivalve resources. Essentially, they overfished the resource. So it's not like overfishing is something new, and keep in mind, overfishing doesn't explain everything, but to the extent that there has been overfishing, it's not a new phenomenon. Okay, so that's the history. And now we go into the early stuff, and I'll start with this. So this is the early to mid 20th century major clam fishery in Southwest Florida. Actually, I think it's safe to say this remains the largest wild hard clam fishery ever executed in the United States or really anywhere. Of course, they're pretty much indigenous to the East Coast, but hard clams are in Europe. They're in California. They're in several areas in the Caribbean, Mexico region. Hard to figure out exactly how they got there, but they are Mercenaria and they are there. We did genetics on samples from Great Britain and California and whatever, and they were all pretty consistent with what we'd find along the East Coast, mostly Mercenaria, not Campechiensis. But anyway, so this Southwest Florida fishery was focused in the Marco Island area. And this bed was giant. This is all information, except for the last two bullets, I guess. Yeah, this is all information directly out of this report from Schroeder. So if you want to see what I'm talking about, you might want to grab this reference. So this clam bed was like 40 miles long and 5 miles wide. That's equivalent to very roughly, you know, Stewart, Fort Pierce, all the way up to Turnbull Creek at the north end of the lagoon, and 5 miles wide. The Indian River probably averages a mile wide. So you take 5 of them, and this thing was massive. Interesting to note that it really was a very tight, and this is important relative to what Brad Peterson's going to talk about with the seagrass connection. These were, you know, the eelgrass, thalassia, or I think it was thalassia. They just call it eelgrass. Common names are always tricky, but where the clams were abundant, there was grass, and these were pretty much mercenary Campechiensis clams, and I'll talk about that a little bit more. But at that time, on the west coast of Florida, all evidence indicates no mercenaria could be found along the Gulf Coast of Florida. So these guys dug a lot of clams, and you can see, I'm not going to read all of this, but you know, they'd be getting 350, 450 baskets a day, et cetera, et cetera. So really the key to this is in 1922, they dug 20 million clams. Keep that number 20 million in mind, because it is a good comparative reference point for the Indian River wild clam fishery and how it compared at its peak. And like I said, these were big clams, and they were mercenary Campechiensis. They'd get about 125 clams per basket, roughly a bushel, not exactly, obviously, but you know, when you're doing clams, you know, little necks, mid necks, top necks, somewhere in that range, even cherry stones, you're probably getting more like 200, 250 per bushel or so. As I mentioned, this was and still is probably the largest clam fishery ever prosecuted. If there was a bigger one, I haven't heard about it. And it did disappear, but there's no clear reason why. It's unlikely that it was overfished, because according to Schroeder, the fishery only accessed a small subset of this 40 by 5 mile area. So, and as you know, it's important to keep this in mind. Overfishing did not drive this population to near extinction. And it's hard to find. If we went out there a couple, 10, 15 years ago and looked around for clams, we couldn't find any, you know, compared to the largest clam fishery ever. So, and this is another theme, is they just disappeared. And like I said, that will be repeated. So that's the Southwest Florida clam fishery, just the beginning of commercial harvest in Florida waters. Okay, so then there was the work in the mid 20th century, late 50s, 60s, around in there, Winston Menzel at FSU, and then Kenneth Woodburn. He was with the, I think, Florida Conservation, which turned into the Florida Marine Research Institute, which turned into FWI, et cetera, et cetera. This is the first known incidence of mercenary importation into Florida, research-based. You can check, I give a good description of this and its potential impacts in this Arnold et al. 2009, if you want to check that. As I said earlier, on the Florida Gulf Coast, there was no prior evidence of mercenary occurrence, including, as I said earlier, evidence from West Coast middens. Whereas on the East Coast, there was apparently mercenary, at least north of Cape Canaveral. How far that goes back, but the middens seem to show that. I talked with Irv about that yesterday. He said, oh yeah, they're getting mercenaria, even in the middens north of Cape Canaveral. South of Fort Pierce, really Campechiensis, and the area in between, which is most of the southern and central Indian River Lagoon, there's really no data there, although it does seem like, again, mercenary were coming out of the middens, but that is not as well established. So like I said, working separately but contemporaneously, these two researchers imported large numbers of mercenary because they were doing hatchery nursery type work up in the Northeast. So that's where they would get them. We didn't have any hatcheries here back in the 60s, although I think Menzel may have cranked one up. And Menzel hybridized them, so that's the first awareness we had of a hybridization under controlled conditions, although I hope to remember to talk more about that. He worked in Alligator Harbor, but Woodburn worked all over the West Coast. He was doing plantings all over the place. Tampa Bay, Cedar Key, Charlotte Harbor, Indian River Lagoon, throughout. So that was a introduction in a lot of areas where Campeachensis didn't occur. This was the first, where Campeachensis only occurred and Mercenaria didn't occur. This was the real letting the genie out of the bottle. And as I say here, once the genie's out, there's no getting it back in. We might be able to reduce the percentage composition of Mercenaria genes in the West Coast population, but you'll never, ever get it out, at least not in any reasonable time frame. Okay, so then good friends of mine, Mark Godcharles and Walt Japp, did an exploratory survey from Pensacola all the way around to roughly Fort Pierce, using these two vessels you see pictured. They did 846 stations. They described no Mercenaria. It was exclusively Mercenaria Campeachensis, keeping in mind these are all morphological descriptions, which are generally good, but not perfect. You can't always, in spite of what you may think, you can't always tell if it's a Mercenaria or Campeachensis just by looking at it, just by the purple coloration, just by the ridges, just by the fat, the width. It's not all diagnostic, and we have learned that by doing genetics and morphology on thousands of clams. And as I said, no Mercenaria were recorded at any stations, but they didn't sample north of Fort Pierce, so again, no information on the Indian River Lagoon. Although they did, and this is pretty well defined in the literature, they did find Campeachensis offshore. And it seems like, at least historically, Campeachensis has been an inshore species along the Gulf Coast of Florida. But when you come around the peninsula and get up towards Cape Canaveral, it starts moving offshore and occurs all the way north to at least North Carolina, if not Virginia, Delaware. And we took a ship out one time, a NOAA ship, and pulled a dredge, and we were able to sample a couple Campeachensis out there and did genetics on them, and they were pure Campeachensis, but they're not like abundant beds. They're just scattered as far as we could tell. Who knows what's really out there, because nobody's ever really taken a close look. So their work kind of supported what had come previously. So then I'm going to talk about the Indian River hard clam fishery. As I say here, growing up, I lived on the Indian River Lagoon and spent a lot of time wading out there as a little kid, and never came across a clam. You know, you can feel them, you can tread and find clams, and that was one approach that the commercial clammers took when they were working heavily in the lagoon. A lot of them didn't use rakes. They just walked around and picked them up in the shallow water. So that's treading, and we just, didn't come across them. And when I would attend the Grant Seafood Festival as a young child with my family, they didn't know anything about clams. It was all oysters. Now I think if you go, I don't know what the situation is now. I've been to the Grant Seafood Festival in decades, but back then, clams not even on the radar. So in the late 70s, hard clams were discovered in Body F, the Grant area. That's roughly Melbourne to Sebastian Inlet in Body F, and it was a major set. You can look at the top plot, and that first set of peaks is the Body F fishery. And it was such a new thing that a couple people, fishermen down there, kind of started coming across them and finding them, and they realized there's money in this, but they didn't have enough capacity to support the processing houses. And there's a lot more to operating a clam fishery than just harvesting clams. You got to get them processed and bagged up. You've got to get them separated in size classes, all the stuff that goes into a wild clam fishery. Of course, it's the same roughly for not quite the same, but similar for the aquaculture industry. They do grading as well. So they actually advertised in New York papers, because Great South Bay on Long Island was a major clam fishery back then. And they advertised in the papers up there, please come down and harvest our clams. So the processing houses have enough clams to make it worth them being in existence. So, there were like 1,200 plus clammers came down, and it was totally unexpected. And that's why I say in that top plot, this is a period of incomplete landings, because the state of Florida just wasn't set up to record hard clam landings. And they didn't get their act together until, and that's not meant to be an insult, it's just it came out of the blue. So we really didn't start getting what we would consider to be good information until like 84, and the clam fishery was dropping off by then. So that bracketed area, 78, 79, up to 83, 84, that was the peak of the wild fishery in Body F. And it was probably could have been the highest peak, even exceeding that second one where I've got 7 million clams. But, you know, we don't know for sure. And then there was this second peak, and this second wild fishery peak was in the early 90s, as you can see, lasted for a couple years, and it was up in Body C, which is the area from 528, just north of Cocoa, my hometown, to State Road to NASA Causeway, which is the bridge that leads over to Kennedy Space Center and the Visitor Center, if you know where that is. And I apologize, I know a lot of people don't know these geographic references, but you know, it's not that critical. You get the general idea. And this one, based upon the landings data, actually was bigger than the Body F fishery, but I don't think that was ultimately the case. Again, I think those early Body F years were the big years. But keep in mind that that peak in Body C, which was another huge fishery, there were over 1,000 clammers operating. You know, the area was covered with clam fishermen. That's 7 million clams, and that Southwest Florida one was 20 million clams in a year. So triple the size. And I don't think that the Body F fishery ever got to that extent. But again, really important wild fisheries, and nobody knows exactly where they came from. But I would say this, Paul Chanley, and I'm going to talk a bit about him. We had a bit of a discussion about this one time. And he said, you know, it would only take 2 clams to repopulate the entire Indian River Lagoon. And I said, well, that's true, Paul, but those clams, one of them can't be in Titusville and the other one in Melbourne. They've got to be close to each other so that when one of them spawns eggs, and these guys are dioecious, and by that I mean they're separate sexes. So if the female spawns eggs, the male has to be close enough to spawn the sperm, because they fertilize in the water column and get those eggs fertilized. Otherwise, they just all fall to the bottom and it's all wasted. So there is a spatial component to this. But again, you know, we don't know exactly where they came from. Just the environmental conditions came together and a huge set. Now, again, I guess I debate with a lot of people, but John Ryther said, yeah, I think it was because we had these rainfall events in the mid 70s, and that led to conditions suitable for clams. I said, no, Paul, we had drought conditions in the late 70s when this set really took place, and clams do much better in high salinity than they do in low salinity. So I would stand by my argument that it was probably, at least for Body F, a high salinity event that created conditions conducive to larval survival and successful settlement, et cetera, that led to the explosion of clams in that Body F region. And then the bottom plot, I just want you to pay attention to that, because that second peak in the bottom plot, that's basically the Body C plot that you see up in the top plot. But the key there is, look, after 2000, the clams just dropped off throughout Brevard County. These are Brevard County landings, and basically they disappeared. So that raises the question, so where did they go? Well, the Body F fishery was done in by extreme rainfall event. We reported on that in this Arnold and Creswell publication, which was part of a larger publication by Diane Burrell and Warren Rathjen. And, you know, so there was a huge salinity event. The salinity dropped to around 13 parts per thousand. Remember that number for a very long period of time. And the clams just died. The clammers, it wasn't overfishing, because the clammers were still out there going full guns. Processing plants were still operating. Then this rainfall event occurred and killed off the clams. They couldn't find clams. The whole thing just shut down, and they moved over to relaying and started working other areas of the lagoon. And by relaying, I mean they harvest the clams, but they're coming out of polluted, quote unquote, polluted waters, uncertified or closed shell fishing waters. So they had to depurate them, either by planting them in clean waters for 2 weeks or taking them into a depuration facility for 72 hours to flush the bacteria out of them before they could sell it. So that kind of kept the clam industry going. And then the Body C fishery, we detected that. We were doing suction dredge sampling out there, and, you know, we hardly ever found a small clam recruit. In fact, we really found practically none. And then one day we were out there, and there were just handfuls of hard clam recruits. And that was the precursor, the first indication of this huge 1990s Body C clam fishery. But again, the clammers were doing great, and all of a sudden, the clam fishery just collapsed because the clam population collapsed. And it wasn't due to overfishing. It was due to an El Nino event, pretty tightly linked in this case, because rainfall lasted for weeks. And again, salinity dropped to about 13 PSU. the clams died, and that collapsed it. And as I say down here at the bottom, the real question is, why haven't they come back? That's the more perplexing mystery, because they haven't. As you saw from that plot, nobody's fishing clams in the Indian River part of the Indian River Lagoon complex, or in the Banana River part of the Banana River Lagoon complex. I think they may still be doing some wild fishing in Mosquito Lagoon. I'm not sure, but there's just really not much going on. So, you know, fishing impacts. As previously noted, the evidence does not support overfishing. You know, you can argue with me about this, but the evidence that I have available does not support overfishing as killing off these two clam populations. But this doesn't mean that fishing didn't have an effect. And that effect may be a very important consideration when you're talking about hard clam restoration, at least in the Indian River Lagoon, where this heavy fishery was prosecuted. Because if you look at this what I would consider a very important study by Marinelli and Wooden out of the University of South Carolina, they did a study on sediment disturbance and its effect on recruitment. And they used Mercenaria as one of their test animals. And what they found was sediments that had been disturbed were rejected by hard clam larvae. So they're not setting in these disturbed sediments. Well, when you've got 1,200 clammers on the water every day raking, and they doing it for years, they've disturbed basically 90, 95, maybe 100% of the bottom out there in that area. That may have created a situation where clams no longer recruit. You can't find, I mean, maybe you could if you looked really hard, but extremely difficult to find a clam in body F, extremely difficult to find a clam in body C. I can't believe there's none out there, but I tell you, I would go out with professional clammers and we'd hunt, at least in body C, and we couldn't find them. Now, again, this was 20 years ago. I don't know what the situation is now, and I want you to keep that in mind, because I haven't worked over there since the mid to late 2000s, 2005, 2010 area when I moved over to NIMS. So, there's still, it may serve to get back out there and take a look if you can find somebody to do it for you, or if you've got the wherewithal to get in the water and get a hand rake and get down there and do the really hard work of looking for clams. So a reasonable hypothesis is that this clam raking intensity, as I mentioned, in the other areas of lagoon, there was raking going on. Even if it was closed, it was for relaying. That may have resulted in inhospitable sediment chemistry composition that interferes with successful recruitment, possibly scents, although there's more to come in the story. And, they're not hard clams aren't the only bivalves in the Indian River Lagoon, and this sediment disturbance may have had impacts on them. But those impacts may not be the same as for hard clams. In fact, they could have been positive impacts to some other bivalve species. Who knows? Nobody's really looked. But that could result in a change in the community composition of bivalves out there. And I emphasize bivalves because they're filter feeders. There are other filter feeders too, but that change in composition can be very important, because it's the implications for doing environmentally focused enhancement could be very serious in this regard. So these sediment disturbance considerations, so like I said, we did suction dredge sampling for hard clam recruits, but of course we got a lot of other bivalves in there. We might not have gotten many hard clams, but we got a lot of bivalves, and in particular, Millennia and Chione. And they were, the Millennia would just pour through your fingers. It'd be thousands and thousands of them in a sample. There must have been, hundreds of millions to billions of Millennia out there. Whether they're still out there, I don't know, but they're a filter feeder. Same with Chione. Chione is a bigger, and some people actually eat Chione. It's a venerid, like hard clams, it's just a smaller one, and they were extremely abundant. In fact, the clammers used to say, we're getting all these small clams, how come they never grow up to be big clams? I had to tell them, because they're a different species. They're never going to get any bigger than that. They're about the size of what the aquaculture industry terms a pasta clam. So anyway, massive numbers of these are filter feeders too, and this brings up the concept of filtering capacity. These, their filtering capacity probably far outstrips that of Mercenaria in the lagoon, even at the peak of Mercenaria abundance out there. So I'll talk about this more, but this idea of filtering capacity is really important to consider with all of the different concepts that are being discussed, you know, nutrient removal, carbon sequestration. It all starts with filtering the water. And the hard clams aren't the only organisms that are out there filtering the water, and they may not be the most important. So did the raking impacts affect these species? And if so, was it in the same manner, or in some way was it differential? And like I said earlier, perhaps some species benefited at the expense of others. It's really, nobody's looked at it. So there's more regarding sediment considerations, but first I want to talk a little bit, this is my last bullet, about the topic of hard clam aquaculture development. So Paul Chanley, I mentioned him earlier. He's the first one I'm aware of that was trying to do hard clam aquaculture in the Indian River Lagoon. There was a lot of work going on, Milford, Connecticut, and all up in the Northeast. Paul came down from the Northeast, and he figured, hey, warmer water, I don't have to suffer through the winter. The clams grow faster. They're just as good, at least as et cetera, et cetera. He never achieved much commercial success himself, but he did set the stage. Particularly, and his footprint remains, as I say, around Sebastian, where I believe, and I haven't kept up, but I believe they still have some successful aquaculture operations, hard clam aquaculture operations ongoing. And what Paul was doing could have contributed to that huge set that feeds directly into what we're going to be talking about in this workshop. Was his, you know, concentrating large numbers of clams in a small area, did that create a spawner sanctuary that pumped out large numbers of fertilized eggs that created the fishery? We don't know. I have no idea. It's one potential hypothesis that we'll really probably never be able to explore. But big time aquaculture didn't take off in the Indian River Lagoon, and it did take off, but not until the body of fishery collapsed, because now you got all these guys, they've got the Chanley background, they've got some knowledge, and people dove into it big time. And they got leases, huge lease areas, just like Cedar Key. They had huge lease areas set aside, and people were putting it out there. And the Florida Today newspaper article back at that time, one of the clammers was interviewed, and he said, "Oh yeah, we're gonna gross $1 million a year from every operation." Well, that never happened, and the question is why? Well, remember I told you about the 90s clam fishery and that wild fishery being obliterated by the El Nino. Well, that also basically destroyed the grow out operations for the aquaculture folks operating in most of the Indian River Lagoon, not so much in Body A at the northern extent, which generally stays kind of high salinity. Mosquito Lagoon stays very high salinity, even in the face of an El Nino. And the Sebastian area, because of the inlet, it tends to stay pretty high salinity. But in between, just any aquaculture operation that was going on was just killed off. And that's what led them, and I'm going to talk more about this too, to get the heck out of the Indian River Lagoon. Although the hatchery folks, and there's several hatcheries operating, they stayed because they seemed to be still successful. Bill Leeming, good friend of mine, I worked with him a lot. His in Body C on the East Bank, he was continuing to go throughout the late 90s when the El Nino occurred. So historically, as we've seen in the middens and everything else, Indian River Lagoon conditions, water quality conditions, environmental conditions support clams versus oysters. Oysters tend to be lower salinity, clams tend to be higher salinity. So as the salinity fluctuates, one or the other species is favored, and that's reflected in those middens, but it's also reflected in the, you know, killing off of this clam population. So also the competition with that 90s wild harvest played a role, as did lease site acquisition issues, and also very importantly, the attitude of local folks around the Indian River Lagoon. You know, these folks living on the lagoon have big houses and lots of money, and they didn't really like having these clam operations out there. So that's another thing worth keeping in mind. It's not always socially popular to have a clam operation going on. Maybe some places, yeah. So anyway, I don't remember when it was, I think it was like 89, I gave a talk at the Florida Aquaculture Association meeting in St. Petersburg because everything was collapsing in the lagoon. And I said, look, you guys, there's a lot of places around Florida where you can do hard clam aquaculture, because we'd sampled and all the history, et cetera, et cetera. And so then the Aquaculture Association funded me to do surveys of the suitability for clam aquaculture, yeah, both in the southern lagoon from roughly Wabasso Bridge south of the inlet all the way down to Stewart, and then that one wasn't real useful, but then also the Cedar Key region. So we surveyed that area and said, here's this subset of the area we surveyed that seems to be appropriate for hard clam aquaculture. And that's really what led to the development of the Cedar Key hard clam aquaculture industry. Nobody had thought about it to that point, but they really bought in. Other people moved to Charlotte Harbor, but they really bought in Cedar Key, and you know, there's the bright shining light known as Clam-a-lot, or whatever Leslie's paper is, really describes all of that. Okay, so then the last throes of aquaculture in the Indian River Lagoon, most of the grow-out operations have moved out or went bankrupt. As I said, around Sebastian in the north area, they managed to survive. And then several hatcheries remained. I also mentioned that, but sometime in the late 90s, early 2000s, the hatcheries ran into trouble, and all of a sudden they could no longer raise larvae to set. They could spawn the clams, but the larvae would die before they set. So I brought Jan Landsberg over from FWI. She's our harmful algal bloom expert, one of them, her and Karen Steidinger. And she diagnosed it as the dinoflagellate Pyridinium bahamense, which has been in the Indian River Lagoon forever, and it was a very benign species. It causes bioluminescence, and we used to love to throw rocks in the water and it would light up, all that stuff, but it wasn't a harmful algae at that time. But as Jan said, it morphed, it mutated into a harmful organism, and it was the cysts apparently that are causing the trouble. They'd go through the aquaculture operation, the hatchery operations filters, and then kill off the larvae. And potentially, you know, I talked about raking impacts, but potentially these cysts that must be all over the bottom of the open lagoon could have a similar impact, and that may be preventing recruitment. But, you know, either way, that's really important if you're trying to restore the population. And again, we'll talk more about that. So I don't know what degree that may have affected wild settlement, but it could have, and that's another important piece of information, not just for the Indian River Lagoon, but for wherever harmful algal blooms may occur throughout the state. Okay, so then we went into restoration enhancement work, and the clammers, whose fishery was basically collapsing, asked me, can you help us by restoring the clam population? And that's when I told them, no, I can't restore the clam population. That's impossible because it doesn't need restoring. And we'll discuss that a little bit, but it could be enhanced, and we'll try, but I'm not promising anything because this is extremely difficult. And as I'll say again, and like I said, I tend to repeat myself, you know, if whatever natural conditions are causing these clams to have trouble, until those conditions change, you can't force something through that environmentally and naturally isn't being supported. So, and my guiding principle on this, as I explained to them, is it's not possible to directly rebuild the clam population. You're talking about, you know, I mean, to the point of having some ecological or economic benefit. That's millions and millions and millions of clams. So my approach to this was rebuild spawner sanctuaries, just like I mentioned with Chanley. His aquaculture operation may have created a concentrated clump of clams that produced a lot of viable embryos. Well, that was the idea. Put them together so they're not at Titusville and Melbourne, but they're all patched together. So when they do spawn, you have a much higher success rate of egg fertilization. So that's what we did. And the other thing I just want to put in, you see this clam, it's important to keep in mind that hard clam life history is not like that of oysters or bay scallops, for example, the other species I've worked a lot with, who are much shorter lived organisms. So hard clam life history, they can go a long time, and I mean decades, without having a successful settlement event. If scallops don't recruit every year successfully, they'll collapse because they're one-year animals. But clams, they can go 10, 15, 20 years without a recruitment event. And then one major recruitment event may carry them through for the next 2-3 decades. So that's why I say restoration is not the term to use, because you're not restoring these clam populations. They may be perfectly happy where they are with extremely low densities, like 1 clam per 1,000 square meters or something, but it still works for them. That's the way they operate. And as I say, a single major set can carry the population for a long time. So Irv gave me this slide of a section clamshell. That clam is 33 years old. He also told me now they're getting clams, I think in North Carolina from middens that are over 100 years old, hard clams. I never expected them to live that long. I thought they might live up to about 50 years, but you get the idea. These guys aren't operating like your most of your smaller and more rapidly growing and dying bivalves. So continued, we took a three-pronged approach to this restoration. The first was transplant adult hard clams. We could still get them up north of Titusville. So I took clammers out there and they worked and worked and worked and piled up 5,000 clams or something. And we planted them into plots. So again, they'd be high density clam concentrations. Our second approach was purchasing clam seed, 2 millimeter, 8 millimeter, and 16 millimeter, also to create spawner sanctuaries in protected and unprotected plots. Separate study, but contemporaneous again. And then a controlled spawn and the release of, working with Harbor Branch, releasing fertilized eggs. They did this massive spawn for me. They got the eggs fertilized. I put them in containers and we rushed up to the lagoon and released them, et cetera, to see if that would work. And we did that several different times using several different strategies. All failed, but for different reasons. The adults, most of them died before the end of the 1-year monitoring period. And keep in mind, having clammers harvest adult clams is very expensive, and it's frustrating to them because they're like, we're harvesting these clams that are worth money and then throwing them over the side of the boat, which isn't exactly what we did. We actually went down and planted them into our plots, but still, they thought we were crazy. We paid them, so they did it, but they thought we were crazy. And there's another thing. There's a gonadoneoplasia that occurs in the Indian River Lagoon that destroys their reproductive capacity as they get older. Now, Pete Peterson will show you from North Carolina, he had a paper that older clams don't get reproductively senile. They get more and more valuable as they get older. In the IRL, that's not the case. And anywhere where this neoplasm occurs, because it destroys their reproductive capacity. So even if the adults were successfully planted and lived, their contribution may have been minimal. Now, when it came to the seed, and you could probably predict this, the 2 and 8 millimeter clams just got eaten alive. The 16 millimeters did better, but they all died within a year. One thing that happened was Hurricane Irene came straight up the lagoon and destroyed everything. We were able to put the plots back together. And the 16, like I said, 16 millimeters did okay, but 16 millimeter clam seed is very expensive. So how far can you really go with that? And then the larval release thing, well, honestly, we don't know. We did a major release. Our biggest one was 550 million larvae, which isn't all that much, in the area between 520 and 528 in the Banana River. It was a nice little 5 by 5 kilometer box. And we tracked the larvae and we did all this really neat science, and it's written up in this Arnold et al. 2005 paper. But we never went back. We didn't have the capacity to go back and see if that's ever set. We surveyed the lagoon with clammers at a different time, with the Banana River Lagoon at a different time. They couldn't find any clams anywhere except up around the turning basin, just outside the turning basin at Port Canaveral. We found one little clump. So my guiding mantra on this, I want to make sure I get this in there because it too is important, an important lesson. If they're not there, there's a reason. So before trying to put them back, it's essential to not only understand why they're not there in the first place, but to ensure that whatever those mitigating factors are addressed. Otherwise, you're just spitting into the wind. You don't do restoration or enhancement in a 13 part per thousand body of water because the clams just aren't going to survive. But if it's filled with pyridinium cyst or the sediment has been upset, you have to take these things into consideration before you start spending large amounts of money to try to rebuild or enhance a clam population. Okay, so just quickly, I expanded the larval release approach. I'm not sure this has any potential, but we did this enclosure, which is a sediment curtain. And there's a curtain that goes all the way down to the sediment, and a chain anchors it to the sediment to minimize exchange inside and outside. And then we released the larvae into there because we wanted to do a controlled experiment. Because when you're doing this thing out in the open, you really need billions of clam larvae. And of course, which hatchery is going to be able to deliver billions of clam larvae. So we created this 10 diameter corral and released the hard clam larvae into there. We had settlement trays down there that we pulled after 8 days or so. We didn't find any hard clams, but we found a lot of other venerids. So things were settling in there, just not the hard clams we released. So the jury is out on larval release. We did a lot of work on this, and we thought, you know, we set up our spawning operation right next to the lagoon, and we thought, boy, if you could put a spawning operation on a barge and just continuously spawn and pour larvae out into the lagoon, that was a thought. And, you know, and just be pumping them every day for as many days during the reproductively viable seasons, because you really can't condition. Conditioning the clams requires algae, is very expensive. We were trying to take a low cost approach, and that seemed to be the lowest cost approach. But, you know, we didn't have the money to do this extensively, et cetera, et cetera. But I'm just throwing that out as food for thought. So the lessons from this history here: clams have been a feature of Florida and its fisheries for millennia, literally for millennia. Evidence suggests the Native Americans may have overfished the resource, but overfishing does not appear to be the cause of the collapse for these 20th century clam fisheries. Instead, it's changing environmental conditions. This is particularly true along the east coast of Florida, but not exclusively. Consider the Southwest Florida fishery. The collapse elicited our studies on population enhancement. Contemporaneously, hard clam aquaculture developed, and how population enhancement dependent on aquaculture techniques can benefit Florida's ecology and economy remains to be determined, and is goal dependent. And no, I'm not done. I would know you wish I was. But so the next step in this is to set up the remainder of this meeting. And here I start with, it's absolutely essential to establish clear goals. As we'll discuss in the following talks, the goals may include one or more of the following. So that would be nutrient removal, carbon sequestration, water filtration, seagrass enhancement, harmful algal bloom management, fishery rebuilding, and potentially others. Maybe there are some other things out there. These are the ones we're going to focus on the next 2 days. So I just wanted to give some thoughts and conjectures on these individual goals, mostly nutrient removal, but not only. I'm using nitrogen as an example. So just getting data from the literature, a 40 millimeter length shell length clam contains about 4 to 7 milligrams of nitrogen. And you guys can, you know, revise these numbers, argue them. It's the basic concept is what I want to focus on here, not the specific numbers. A commercial clammer harvests around 1,000 clams per day. Bill Leeming would get 3,000, 4,000. The guy he was a phenomenal clammer, but your average clammer is going to get around 1,000 a day. So what's that? That's 4 to 7 grams of nitrogen per clammer per day. So using the 1994 peak, that's 7 million clams, or using the 1,200 clam fishermen, that's estimated to be about 11 pounds, 5 kilograms of nitrogen removed per day for a several-year period. The EPA estimates is about 3 billion pounds of nitrogen per year into the lagoon, of which about 1 million pounds is excess. So estimating 250 fishing days and 11 pounds per day per clammer, that's about, well, 11 pounds per day total for the fishery. That's about 2,750 pounds per year, or about 0.25% of that excess of the 1 million pound excess, not the total, but the excess. So that's an important context. Is this really worth, is this having enough of an effect on nitrogen in the lagoon to make a difference? These are questions, not answered. This is the kind of stuff that needs to be explored. And this is dependent upon harvesting those clams. If you just leave them out there, they're going to die. And at least to some extent, the nitrogen is going to be recycled right back into the water column. Not all of it, maybe. And we need studies on what are the dynamics of nitrogen in and through clams. And I think Ashley's going to talk about that more tomorrow. So, you know, the wild fishery is gone. And as I said earlier, it's not just the fishermen. You got to have the processing houses and the markets, et cetera, et cetera. So if you're going to use hard clams and your goal is to remove nitrogen by growing the clams and removing them, somebody's got to remove them, and that's a big job. So you got to have a fishery. That fishery, of course, is going to compete in the market channels with the aquaculture industry. So that's another thing that has to be considered. Is the market big enough to conduct a nitrogen removal operation? Because if you're going to harvest the clams, you got to sell them. That's the crux of this whole thing. And that's what makes hard clams distinct from all these other clams. I'm going to talk a little bit more about that. So finally, it's very important to keep in mind that the aquaculture industry, and particularly the hatcheries, may not have the capacity to produce adequate seed to run both an aquaculture industry and a restoration industry. When my team obtained the fertilized larvae from HVOI, and I thank David Vaughn and Leroy Creswell and Richard Baptiste greatly for their incredible support of us in doing this work, David said we blew out his broodstock, and that was, you know, for them, that broodstock is gold. So, and that was just 550 million embryos. As I said earlier, billions will be required. So is the capacity there? And I don't know, but that's another question that has to be addressed. So on to carbon sequestration. This is a shell concept rather than a soft tissue concept. The shell is roughly, this is my guess, Patrick can correct me, my guess is 75% of clam weight. It certainly is the majority of clam weight. And you don't need to harvest them because it's sequestered in the shell. So as you saw from the middens, these shells stick around for thousands of years. So once the carbon is sequestered, even out in the lagoon, even if you don't harvest them, it's going to subside into the substrate, and pretty much the carbon's going to be sequestered again. I looked to Patrick for the details on that, but that's a rough overview of it. So carbon sequestration is a more straightforward goal to achieve. And again, I'll leave it to Patrick to discuss that in depth. But you still have to have clams out there, and you know, there's no magic. They're not just going to appear. So then the water filtration goal. This is somewhere between nutrient removal and carbon sequestration. Maybe you don't have to remove the clams, but you need the clams to be alive and cranking. They can't be dead and the shell subsiding into the substrate. So sediments are removed from suspension, and they may be deposited on or in the sediments via pseudofeces or integrated into the soft tissues of the clams. And there's a whole, you know, recycling thing there, but you're removing sediments. But some of that may be recycled upon death, and that's what Bruce will talk about. So, you know, as I said, I'm kind of setting these things up here. So seagrass enhancement, Bradley Peterson's going to talk about that, and it may be species dependent. As I said, the Mercenaria campichensis seem to be pretty closely associated with seagrass. The Mercenaria are not. So take that and keep it in mind. And also, you know, I did a lot of work with habitat growth relationships of these two species and their hybrids in the lagoon. That's the Arnold et al. 96 report. And it's not all, they don't all just occur equally in all the habitats. They don't grow equally in all the habitats. Each species, this is how we maintain this hybrid zone in the two species. They have advantages in different habitats. So you got to take that into consideration when you're talking about seagrass enhancement. That's not, again, not to say that it's not a viable approach and a useful approach, but there are many factors that must be accounted for. And then harmful algal bloom management. Well, like I said, in the Indian River Lagoon, at least when we were out there, pyridinium was winning, not the hard clams. You know, it's been shown adult hard clams can filter red tide, Karenia brevis on the West Coast. It doesn't seem to bother them much. But that doesn't mean that they're recruiting. That doesn't mean that the larvae aren't bothered. That doesn't mean that they can successfully settle into a pyridinium infiltrated, you know, cyst infiltrating the substrate all over the place. Maybe that would prevent recruitment. So whether you can manage harmful algal blooms or not, and it would depend upon the species of HAB, that all needs to be still evaluated. But as I say here, one size does not fit all when it comes to harmful algal bloom management. And finally, fishery rebuilding. Well, this is an interesting topic, but this is what we were focused on. And as I say, it's a lot more than just putting clams in the water. And putting clams in the water is a hard enough job. But you need the fishers, you need the infrastructure, the processing houses, et cetera, because otherwise fishermen don't have anything to do with their clams. You need greatly expanded markets to accommodate both the aquaculture and wild harvest industries. And keep in mind, if you're dependent upon the aquaculture industry to drive your fishery rebuilding, well, okay, then they're going to say no if it's just putting them out of business. And as I said earlier, you need community support. And if you don't get community support, at least you need community acceptance, because when they start going to their county commissioners and saying, get these clammers out of my backyard, the clammers are going to have a problem. And that can apply to a lot of different components of the aquaculture wild harvest, because they don't want clam leases in their backyards either. So you got to take these things into account. All right, so keep in mind, as you probably know, all of these goals depend upon filtration, except perhaps fishery rebuilding. As I mentioned earlier, Mercenaria are not the only filter feeding species in the Indian River Lagoon or in any other lagoonal estuarine environment throughout Florida or the US. When we're talking filtration capacity, there's nothing special or almost nothing special about hard clams. And in fact, there are aspects of their life history that can mitigate against using them, against their value, if you're trying to achieve these conservation goals. I think this is something really important that hasn't been well considered, because these chiones and millennias, they grow a lot faster. They probably, and I don't know this, I don't have the evidence to back it up, but they probably metabolize at a much higher rate, which means they would be far more effective. But nobody's going to harvest chione and millennia. If you need to remove it by harvest, hard clams have an advantage. If you're just going to subside carbon into the substrate or, whatever, you don't need hard clams. These other species may be a better choice. So like I said, mitigating against hard clams are very long life, 20, 30, 50 years, 100 years. They don't turn over real quick. And, they may have a highly unstable population structure. You can get 10 million of them out there, but in 3 years, you may only have 5,000. And after they grow rapidly, their whole life history is to escape predators at a small size by growing rapidly and outgrowing their predators' capabilities. That was my first publication doing my thesis work at Skidaway, is looking at size-dependent predation of blue crabs on hard clams. So they get big fast and then they slow down and they hardly grow at all, which means they're hardly metabolizing at all. They put most of their effort into reproduction. In contrast, turnover rates of chione and millennia, among many other bivalves that are out there filter feeding, much more rapid, so they may serve these goals much better, especially if achieving the goal does not require removing the clam shoreside. So these, again, need to keep this in mind. And there may even be other phyla that are out there filter feeding, like polychaetes. So in summary, I provided a brief review of clams and clam research in Florida. And I always say the first job of a scientist is to build upon previous work. That's what I was trying to do with the history I gave, and that's what people coming now and going forward need to do, is be aware of and build on what's been done. If you're not aware of it, if you're not aware of history, you're destined to repeat it, probably in a very expensive and unsuccessful manner. You need to establish goals, and then you need to develop an approach that will achieve these goals in a cost-effective manner. Remember, we're talking about taxpayer dollars in almost every case, so be respectful of those taxpayers and their money. You need to provide a context I tried to provide a context for evaluating the realistic potential for enhancing hard clam populations to achieve these ecological or economic goals. And as I said, I'm hopefully setting all this work, and a lot of people doing a lot of hard work, missing a lot of important meetings and memos back at the office. I hope this sets the stage for future research needs. And as you can see, my opinion is there's a lot of research needs that need to be addressed. So I hope this information proves useful in the discussion that follows. And as we move through the next 2 days of the workshop, maybe so, maybe not. I'm sure many of you may not agree with some of the things I've said, but maybe it'll stimulate you to get out there and prove me wrong or to advance the science. So that's my talk. Sorry it went so long, and I'm happy to entertain questions.
Wonderful, Bill. Thank you so much. That was a ton of information. I feel like I just breezed through a book.
Well, there's more. I couldn't present every piece of information we ever obtained. I tried to hit the high spots as they apply to this audience.
No, this is wonderful. All the information that you've shared it's quite important to really understand that background and everything that we need, especially to move forward. So thank you for taking the time and sharing that with us. I have recorded some of the questions that have come in during your presentation, so I will go ahead and ask you those. If anybody else in the audience, the participants, please feel free to go ahead and comment on the chat box if you have any more questions, and I will make sure I will get to those after I go over the first few questions that we have. I'm sure, Bill, if we were all not unmuted, you'll hear lots of applauses and thank you. So I will start with the first question and I'm very sorry if I butcher your name. I'm going to try my best to not. Jesse Wales, ask, was Honeymoon Hill taller than Turtle Mound in Canaveral National Seashore? I suppose she's talking about shell mounds, or...
I know.
Okay.
So I've never been to Turtle Mound. I'm very familiar with it. Irv and Doug worked there a lot. Honeymoon Hill was easily 30 feet tall, maybe 40 feet tall.
All righty. Wow. I can only imagine 30 feet. All righty. We have a question from Steve Geiger. Did you ever document density of other species or only clams? I suppose I think he did send this question when you were talking about the Indian River Lagoon, but feel free to comment on your other work as well on Southwest Florida if you did some of that.
Well, we didn't. For one thing, the millennia are too small. We would do meter square quadrats and we would hand rake them for the hard clams. It's a great sampling method for adult hard clams, not recruits, but for adults. And that was our major sampling for the adults. And then we did suction dredge sampling for the clam recruits. Even the raking didn't even get really Chione, which are about, like I said, 3 to 4 centimeters in maximum size, whereas the millennia are about 1 centimeter. When we did the suction dredging, we would bring up gallon Ziploc bags full of millennia. Now, we didn't quantify that. There were only 2 of us, Dan and I, and we had to do the clam work. That's what we were funded for. We were using hard clam license fees. So I would just say that in a square meter or in a quarter square meter plot, which is what the suction dredge sample frame was, there would be hundreds of millennia. But keep in mind, this is really important to keep in mind, and I said this earlier, the IRL has changed a lot, and not for the better, since 2010, like in the last 10 years or so. I don't know the exact timeline. So I don't know what's out there now. All these, you don't find hard clams. You may not find Chione. You may not find millennia. I think it might be worthwhile to take a look, but no, I don't know. Maybe Brevard County Natural, their natural science office, they used to have that. I don't know if they still do. And a guy named Ray Grizzle used to go out there and sample. Oh, and I can't remember the other guy's name. They were there forever, and they had good data back in the 60s and especially 70s on all the species, because they were doing, community composition type stuff. But recently, I don't know if any of that stuff exists. You just have to get out there and look. Things change. I think change constantly in a biological environment. Oh, and sorry, just to finish that up, we did that sampling in the late 80s, early 90s. So it's been 30 years.
Gotcha. Well, that's a great baseline anyways, that you're providing us with, like really understanding. A lot of us working currently with the current conditions and really thinking what it was back then. It's just really a great information to have that from you, because then you can really compare, through all the changes, environmental changes that we know have occurred in our estuaries. It's quite nice to know what it used to be like, which a lot of us, want to restore to historical conditions, but what was that really? What are we really aiming for? So this is... And
I'm sure what we sampled was a lot different from 1910 or 1218 or whatever. But I can't believe there's not some bivalves out there filter feeding throughout the state of Florida.
Absolutely. All right. Our next question was by Ed Sherwood. He asked, can you comment on any potential changes in estuary trophic structure that could affect clam distribution? That's one question. And he followed on, we now observe large fevers, sorry, it's a word I'm not used to, of rays each year along Florida's Gulf Coast. Are top-down trophic structure changes now affecting clam populations in Florida?
Well, physical, chemical, and biological factors, and even to some extent geological factors, can all influence community composition, including where hard clams fit into that community. And I think it's very worthwhile for folks to keep in mind you're operating within a biological community, not within a biological population. You're trying to enhance. My take on this is, and this is clam restoration, not hard clam restoration, but from the point of view of hard clams, you are looking at a population and trying to enhance it, but nothing happens in a vacuum. That community is very important to keep in mind. And that community, from an ecologist point of view, includes the seagrass beds, for example, the sediment composition, how sediments were disturbed, as I discussed earlier. Where does pyridinium fit into this? What else has changed? Grain size is important, but also, the stingrays used to be extremely abundant in the Indian River Lagoon, not cownose rays, but southern stingrays were very abundant. Whether they're moving through the lagoon or not, I don't know that there's some drastic increase in the number of cownose rays. I know Peterson did work on cownose rays predation on bay scallops in North Carolina. It was an interesting study. It was published in Science, but I think if you read it closely, you'll realize there's some flaws in that study, and you need to be careful about how you interpret these things. I know that the cownose rays hit the aquaculture bags pretty hard, at least some of the time, but whether they are hitting 5 millimeter, 10 millimeter wild set clams out in the open lagoon or not, or the open estuary, I don't know that. I imagine they would if they came across them, but it may be they like clams because they're packed in at such high densities, they can get tremendous return on their feeding investment. Whereas if you're looking at scattered clams, you're putting a lot more effort into search and a lot less effort into eating, and energetically, it may not work for you. So I don't know if that answers all of Ed's question. I know he followed with a second one, but I'm trying to answer that by saying community composition is absolutely essential. And you need to think of that community composition in the largest sense, not just what the other bivalves are, but everything, because lots of things will prey on a hard clam larvae that's trying to find a suitable place to settle from the pelagic into the benthic environment. A small worm can eat a 250 micron setting clam larvae.
Thank you, bro. Yeah, and I also thought that was very insightful and lots of great information that we must consider as we move forward with these restoration efforts. And I also want to point out, and I know that Angela Collins did this, but mention it in the chat. I know that Matt Ahemian, and I'm sorry if I butcher your name, I know he is, I think, working in Florida Atlantic University, and he is going to be briefly talking about his he has a project with Sarasota Baywatch following up a restoration project looking, I think, is mainly like predators, so that focus on clams. And so I think he's going to be mentioning a little bit of that. So I leave that to him in our following up group discussion. So perhaps he might have some information for us a bit more on the ray side or those predators that are very clam focused. All right. So next we have another question by Vincent and Comio, and he asked,
Hi, Vince.
he asked, was the work on pyridinium cyst by Jan Landsberg published? If so, can this be posted here? And I believe maybe Angela posted that, but if not, Bill, feel free to comment.
I don't think she published anything.
Okay.
I mean, we couldn't get enough data. The clams are all dead. You need to do a, clam controlled study, and there weren't any larvae to work with. And putting clam larvae in with pyridinium resulted in a very rapid mortality event.
Gotcha. All righty. We have another question by Jay Leveroni. What's the current status of the Marco Island clam beds?
As far as I know, we went out and looked, but this was probably 20 years ago. We couldn't find a clam. And you're talking about what must have been, I mean, they harvested 20 million clams in a year. And I know I'm old. I wasn't alive back then. I don't know exactly what went on, but you saw from Schroeder, he said 20 million, and they were just harvesting a small patch of the total 40 by 5 mile bed. So the number of clams out there must have been phenomenal. Now, Schroeder did mention that, and he didn't go into depth, and it didn't really explain things. But he said there was a big freshwater burst coming out of the 10,000 ounce. That was back then. That was the drainage. Water wasn't cut off by Alligator Alley, and it was still flowing down and sheet flowing out of there from all of Central Florida. The Everglades was operating as normal. So I don't know, but now, you know, you could go down and look. It takes some hard work, but God, Charles, and Japp didn't come across any massive. That'd be where I'd start. I'd start with the God, Charles, and Japp publication and see what they found. I don't recall, I don't have the whole pub memorized, but I don't recall that they found any extraordinary number of clams down there. And it must have been an extraordinary number of clams. And if you look at the geography of that area, you come around Sanibel Island, and then the coast goes way in, and that's a recipe for gyres, creating gyres. As the current that comes north to south along the coastal current comes north to south, it's going to spin in, and that gyre could have created a situation where larvae are retained, and that would create a really good recruitment opportunity. Maybe that gyre moved a little bit, or you just don't have the brood stock, or there's a million different explanations, and I can't tell you what they are. All I know is I don't know of anybody harvesting commercially down there. And so I don't think there's anything substantial. I think if you went out there with a clam rake, you'd have a hard time finding a clam.
Gotcha. Yeah, and I just saw a comment by Leslie Stormer who said the clam aquaculture leases established in the 10,000 islands in 2002, 2003 were never productive.
Yeah.
So, alrighty, how are we doing on time? Let's see. Alrighty, we have a few more minutes just to wrap up the questions. We have one more question. Let's see. Nano Riley asked, are there razor clams in Florida, and where? Do you know, Bill?
I believe so, and they're in more high salinity environments like along the beaches and stuff, I believe. I wouldn't claim to be an expert on that. You could go to the Sanibel Captiva has a museum. Always Bill Lyons knows everything about mollusks in Florida. The Florida Museum of Natural History has an excellent collection. But, you know, a lot of razor clams in Georgia when I was working up there. But that's a high salinity environment. I think they're higher salinity. I see Steve says they're just small species. So I do think they're out there. I don't recall coming across many of them in our suction dredge sampling, but it was a long time ago, and I don't remember everything that was in those suction dredge samples.
Gotcha. And our last question for now is by Sugandha Shankar. Is there a report, a reference to your observation of impact of pyridinium cyst on larval recruitment?
No. Again, there's a research opportunity. There's a lot of research opportunities. Me and my small team, a relatively low budget, they couldn't possibly tackle all of the many questions that we had. And we, you know, nothing like being out on the water to develop questions. You're out there thinking, and you look down there on the bottom looking. You know, this was sitting on the, you can't see in the Indian River Lagoon. You can't see your hand in front of your dive mask. So you have to be down there operating by braille with a hand rake and a suction dredge. You got hoses all over the place and mud's pouring down on your head. And when you're doing quadrat raking, you know, you're getting stuck with old stingray barbs, and so it was hard work. It may not seem like it should have been hard work, but it was really hard work, especially in the middle of the winter. And I know this isn't 12 miles to school both ways, uphill through the snow, but you know, in the Indian River Lagoon with a northeast wind blowing down that lagoon and the temperature is 45 degrees and you're diving, you're cold.
I bet you are. I'm always cold here in
Florida. A lot of
Florida.
time to think and not a lot of time to pursue every thought that comes to mind. And I apologize for that.
No, but that is wonderful, Bill. And I think that's why we, you know, it's like the saying of stand on the shoulder of giants. You're opening up all these great ideas and things that we need to really know more. And that's why we're all here. And there's lots of science happening. So I think we can definitely build upon on all of those good ideas and questions.
So Dinorah,
yeah,
I appreciate you saying standing on the shoulders of giants, but remember, that's what I was doing. I didn't come into that with no background, no foundation. I had learned from a lot of really good people, including my mentor, Randy Walker up at Skidaway Institute of Oceanography, but Irvin Doug and, all those folks doing work up there. So, if I didn't know the literature, Sandy Shumway was going to crawl all over me because she's our editor at Journal of Shellfish Research. And that's her mantra is my mantra. You better know the literature. Don't start your research project if you don't know the literature. And if you don't, but you do it anyway, don't come to her because she'll rip you. She'll just rip you.
Great advice. Great advice. All righty. I think we're just going to stop here for the questions. Feel free anyways to keep on popping up questions as we go. We're going to take a 10-minute break. Right now it's 10:42. So let's reconvene at 10:52. Just grab your coffee or water, bathroom break. We'll be back, and in a way back, we'll be discussing with some of the groups that we know that are doing restoration here in Florida and learning a little bit more about their efforts here. So we'll see you back in a little bit. Alrighty, and we are back. I'm not sure. Am I sharing my screen? Let's see. No, I don't think so.
Yeah, you are. I
I
see
am?
the 10-minute break. Yeah.
Okay, I think that might be the virtual producing team, which is totally fine. That's great. Now, well, we just heard great history of clam restoration and goals of restoration from Bill. So thank you again for those for that recollection, and also for advice for all of us who are coming into the field and moving forward in that direction. Lots of good information and to learn. And now we're going to move into the group discussions. So this portion of the meeting is to invite all those groups that have been doing restoration or planning to do restoration projects using clams here in Florida. If there's more participants than the one I call, please feel free to, after we go over this, the first few groups, feel free to go ahead and let us know about your project as well. This part is going to be slightly similar. We'll just go over each of these groups, and then they'll give us a brief description about the projects, how they came across this, or what are they doing, what are their goals, what are their plans, and also after each group finishes their overview, if you have questions, please feel free to go ahead and type it, or I guess go ahead and speak it out loud so we can have a bit more of a dynamic conversation at this point. So first I want to start by inviting the group from, there's a couple of groups from Southwest Florida. So first I want to start by, I think, the Charlotte Harbor area, and I believe that will be the group of Sanibel Captiva Conservation Foundation. And there's multiple people involved in here, so I'm just going to mention them all, and you guys let us know if you have any slides that you want to share. We will give you access to do that. And if not, that's okay. We just share with us any information you'd like us to learn from your project. So we have Eric Milbrandt, Leah Reidenbach, Mark Thompson, and also Melinda Brown. These are all the involved in Charlotte Harbor different groups. So take it away.
Hey, good morning, everyone. We, I guess about a year ago, were given some money from the local shell club to do a project that involved a pilot study looking at the mortality and feasibility of doing some kind of an enhancement project in San Carlos Bay. That project is being led by Leah Reidenbach, who I believe is on the call. Mark Thompson could not be here, but he and I and Leah also around October of last year or so started working on a habitat suitability model for Charlotte Harbor for Mercenaria, using a lot of previous data and a meta-analysis combined with 10-year stratified random sampling effort that has been coordinated by the CHNEP, the Coastal and Heartland National Estuary Program. So we were able to produce maps of suitability based on a variety of things. Maybe I'll try and find the table that we use. I don't want to spend a bunch of time looking for stuff, but I think I can find it pretty quickly. Let me share my screen. Okay, it doesn't look like I can share a screen unless Dinorah.
Yep, 1 second. So we're working on that right now. Let's see. All right, see if that works.
Screen, yep.
Awesome.
So if you can see that, we used these factors, salinity, and provided a range of different values, seagrass coverage, flow or fluid flux rates. The flow rates were based on NOAA tide estimates, substrate, chlorophyll, whether it was a harvestable area or conditional harvesting, depth, dissolved oxygen, and temperature. And Mark used basically, the model is essentially a weighted spreadsheet type model in GIS. If I had to describe it, that's how I would describe it. So let me show you, scroll down to some of the final maps. That's leases. Okay, combined suitability factors with the scores. So the score, the value for the score doesn't really, those numbers are meaningless essentially, but it's a combination of those factors I just talked about. And the red areas are areas where there's seasonal hypoxia that's in the data, the 10-year data set that we use. So we categorize those as unsuitable. And then we also did, in the middle, of course, of this last cold January and this in December, we did sampling at 10 sites using raking, some coring to look at juvenile bivalves, and to see in those 10 sites what that were designated as most suitable, what the field conditions were like. And I can share this report with you all. We submitted it to our colleagues at the Charlotte Harbor Aquatic Preserves Office, which helped fund the work. And we were happy to find Campechiensis at a number of those sites. They were adults and a few juveniles. And we also did find quite a few Chione, as Bill mentioned before. So as I said, I'll share this report with y'all. Happy to entertain any questions. The day that I was raking, the water temperature was 57 degrees. So, so that was great. If Leah, if you're attending, if you want to click on and talk a little bit more about the pilot project that we are planning using adult clams and monitoring their progress.
Sounds great. Leah, you were referring to Leah, right?
Yes.
Leah, are you present? I'm gonna look, but there's 83 of you.
Yeah, is it Leah Riedenbach?
Leah, there you go. Sorry
Yep.
if I'm.
No worries. I've asked her to unmute. I'm not sure if she's able to do so. It looks like she's on her phone.
Okay. Yeah, she's having audio trouble. I just saw a brief message on the chat box. She said she'll come back in 5 minutes. It's okay. We can come back and figure it out. So we can continue. So I have a question. So you said that's great. You guys have a report. So I think it will be really nice. We're putting also like a folder with all this information and references and things like that I think will be helpful for the community to know. So we will love to have that document for sure available to share with everyone who's interested. And so those maps that you guys came up and the condition index that's like, or how did they overlap with some of the, I guess, farm leases that are already there? Is that like, do those farm leases fall into like the good habitats, or like was that an interesting finding? What did you guys find about that? You just muted yourself, sorry.
So most of the lease areas were in the highly suitable designation, there was a few areas that were just a little bit below, and they're in high salinity areas with no seagrass. So those were kind of based on a lot of the assumptions that were made were based on where we could potentially have a spawner sanctuary area that the clam farmers could either designate their existing leases as sanctuaries, and some of the farmers that we talked to are on the call, or other potential areas that we could get designated by FDEP aquatic preserves to do spawner sanctuary. So we didn't want to put them in seagrass, for example. We wanted to use areas that weren't open to recreational harvest. So there are a bunch of things that you'll read in the report that were our assumptions in that.
Very cool. And so where does that effort stand now? Now you have this really awesome map that provides you with really good information. And you know, what about, like, are you already restoring, or where's the process in there? Like, what's happening?
So that's a great question. I think there are discussions that we're going to have with the farmers about you know, possible funding sources to do that. We have some permitting hurdles that we need to get over as far as, do we use existing leases, or do we you know, try and establish new sanctuaries? And then the other thing, of course, is the methodology for establishing them. Do you could use mesh as they do, as the farmers use mesh, they really feel strongly that that's an important part of it. And so there are a number of things that we need to continue to discuss in order to really move forward with any kind of larger scale project. But Leah, who's in my office, she's going to sit down and tell you a little bit about a small pilot project that we're going to conduct in San Carlos Bay with a lot of monitoring of both the clam size and the reproductive status. So hold on, she's going to sit down.
Not a problem.
Oh, where?
It's in your email.
Okay, I'm just going to share my screen one more time. Leah's standing right behind me, and she's going to sit down and share. Thanks.
Hi everyone, sorry about that. I just had a little trouble with the audio on my computer.
No problem.
So the first thing is that, well, I was basically inspired to do a hard clam restoration site because I actually came down to SCCF up from Long Island, where I worked with Brad Peterson, and he does a lot of hard clam restoration too. So I kind of just brought some of my ideas about that down here with me. So we applied for a really small grant just to do a pilot study to see if hard clam restoration can even work in this area, because I don't think anybody's ever really attempted it before. So we're going to basically be taking 12,000 hard clams and putting them in a small area, and we're going to monitor the survival, reproduction, and water quality for 1 year. So that includes looking at their gonads, seeing if they're sexually reproductive, checking the sex ratio and gonad readiness, so we can kind of understand how reproduction in this area works, since it's actually different in different parts of their habitat. So we're going to be monitoring the water quality over this small area where they're located, see if there's a change in community structure. And we're going to do that using a hard clam, or not a hard clam, a flow cam. We're going to be measuring water quality in situ, and we're also going to be looking to see if there are changes in nutrients over the area where the hard clams are present, because we know there's still a lot of questions regarding their nutrient uptake ability. Eric already covered the habitat suitability model with you guys, which is going to be a great way to guide us to where clams can potentially survive. So we did do a preliminary survey of the sites that we were interested in. And we found that they were kind of muddy sand areas with some tube worms. We didn't actually find any adult hard clams, but we didn't use, we just did a visual inspection. We didn't really use hard clam rakes. So that is kind of a new method that we're learning is really effective for looking at for adult hard clams. We didn't really see too many predators at the sites, and there was some seagrass in the area. So we think that these are really good spots for them potentially. But personally, I have seen hard clams nearby in that area when I've been snorkeling. So I think that we found some good potential sites for restoration. This is just showing you some of the technology we have for looking at phytoplankton community structure. We just put samples through the flow cam and it takes pictures of them. And then we can kind of see which genre are the most, most abundant and if they change over the hard clam area. So this is kind of some previous work that an intern did looking at different sites along the Caloosahatchee River. So you can see that we can get results using this flow cam. And we plan on starting this next week. We're working with partners from Sarasota Baywatch. They're donating the clams to us since we have such a small budget. So let me know if you have any questions.
Wonderful. Thank you, Leah. How do you say your name again?
Leah Reinbach.
Leah. Okay. Sorry about that. Thank you so much for sharing that. Lots of good info. I also have some questions because some people are looking at you at the same time as the presentation. And I think they want to see a bit more, like a bigger screen. But is it possible afterwards to perhaps like have a copy of the presentation or something that we can just share with the participants if they just want to take a look a bit more in detail? We can definitely put it in a Google Drive folder along with all the other reports and information if you feel comfortable sharing that. Wonderful. We have a question by Steve Geiger. He asked, do you see zooplankton in the flow cam, in particular bivalve larvae?
Which say?
She asked if you could see bivalve larvae in the flow cam.
Yeah, we can see bivalve larvae, but it's very difficult to identify what the species is. So we're still working on that. So we have big enough flow cam, what is it, like
The flow cell.
Flow cell that we can look at larvae, which is probably going to be something we look at in the future.
Very cool. Let's see. Eric, there's a question for you. Are you aware of Bill's publication on lease site selection in the Pine Island Sound area?
Yeah, absolutely. We used all the literature that we could find, and that was a huge part of what we did.
Wonderful. I think there's a question for Leah, and I'm sorry again if I'm saying the name of it wrong. Are you only monitoring for 1 year? And if so, why did you choose this amount of time? By Carolina Panos.
The main reason we chose 1 year is just because of funding and how often we can take the boat out to go to the sites. And yeah, that's pretty much it. Just logistical constraints. It's a small grant.
Yeah, I can imagine. Always, I think that's one of the main things that came up in the responses for the survey is like funding, lack of funding. You know, we need more data, and of course, there's that issue of funding. So totally.
Results from this project can help us just get more funding to do bigger projects or keep monitoring the site that we have in the future.
Wonderful. Nice. Okay, cool. And this is also in the Charlotte Harbor region, right? That you're doing this pilot. Is it ongoing right now?
No, we're going to be starting probably in the next month or so.
Very cool. Nice. Very nice. Well, thank you guys. I don't see any more questions, but I do know that there was someone in the audience, Angela Collins, told me that I think it might be a farmer.
Yeah, can I talk for a sec?
Yeah, go ahead.
Yeah, no,
I
I just think it's relevant because we're talking about the Charlotte Harbor area. So Tyler Heave is one of the shellfish farmers in Southwest Florida, and he has leases in both Charlotte Harbor and also the 10,000 Islands area. And one of the oldest shellfish farming families in the state. And these guys actually are pursuing actively restoration activities of native clam beds in Charlotte Harbor. So I thought since he's on the call, he might want to just chat a little bit about some of their efforts and let the group know what they're doing down there. Tyler, can you hear us? If you're on your phone, press star mute. Oh, there you are. Yay.
Good morning, almost noon. Would you like me to go ahead? Let's just start.
Yeah, I think that you might know some of the folks down there. So anyways, just take it away.
I've been around for quite a while. Again, my name is Tyler Heed. I'm with TurnOurTides.com, and I have 25 years experience in the aquaculture industry. Now, 20 of those years were specializing in industry standard bottom planting techniques. And we like to utilize native species, get better results. You know, industry is all about financial efficiency. But we do have test beds for different techniques, you know, R&D throughout the years. And today, our oldest living test bed, using Campachensis, as far as we can tell. We did do a couple genetic studies with Huaiping a few years ago. That bed, which I just recently checked on last month for a video idea, is 16 to 17 years old and still thriving. I did see mention of, I believe it was in the comments about nobody working in the 10,000 islands. I also have a wholesale division, cutthroatclams.com, and that purchased from farmers down in the 10,000 islands area, and we ourselves have leases down there with a successful Sunray Venus clam crop right now in 10,000 islands and in Pine Island Sound. So that's pretty much it. We just went back to basics for production, but it can easily be tweaked because the devil's in the details, and it is hard. It can, you know, it can easily fail. Once they get up to a certain size, you can take all the man-made predator prevention off, and you're left with a sustainable, self-defensible clam bed, utilizing native species.
Very nice. Thank you so much, Tyler, for sharing that information with us. I think this is like one of the main goals of this workshop is not only to digging into each of the ecological services, but also to really establish that connection and communication among the different groups, the farmers, the industry, you know, the scientists, everybody who's interested in this topic. And this is great. So thank you for jumping in and letting us know about that. We will definitely keep in touch for sure.
Thank you.
Yeah, thank you, Eric, also, and the Sanibel Captiva Group Conservation Foundation. If you guys don't have anything else to add, which you, of course, you share a lot of really great information as well, I will go ahead and wait. Did you say you were doing a pilot study with Sarasota Baywatch, or that's the one where Leah talked about, which is likely that one?
Yeah, that's the small. It's yeah, that's the one Leah talked about. Thanks.
Absolutely. Okay, thank you so much. All righty, moving on. Now we're gonna hear from a little bit north, Sarasota Bay. We have Sarasota Baywatch, who I know that they've been doing several clam restoration projects, not only with clams, but I think previously with scallop. And I think I'm going to invite now to Ernesto de la Vega, Lazo de la Vega, to share with us a little bit of that. If you will, Ernesto.
Thank you, Dinorah, and thank you for everybody for attending. And I'm very humbled to represent Sarasota Baywatch because they have been way before I started with the group doing the work. If I may share, I'm requesting to share.
Yeah, try it out, Ernesto, and let me know if you need more.
Yeah, in fact, I'm going to, and it's very short, so because I want to save enough time for everybody to give a presentation. So
not a problem. Go ahead, Ernesto. Take it away.
All right, thank you. In fact, playing here with the. Okay, I hope you guys can see that. If you're able to see Sarasota.
Yes, we can.
Oh, great. So this is a project that has been for years. We have been doing other things. Like first started with scallop, the base scallop Argopecten. We were doing that at the beginning. I mean, we're talking in 2017, we started releasing clams. But they were environmental challenged because the salinity changes, low oxygen, red tides, all this stuff put a burden on this. So we switched from a scallop to the clams, to the hard clams, the Mercenaria campechiensis. And we saw an excellent opportunity to use this because of the tolerance of the changing conditions, the great filtration they have, the lifespan of 30 years, my goodness. So we saw this as a better potential organism to do these kind of projects. And the goal, and in fact, I'm so glad to hear Bill mentioning it, we create filtration cells. That's what we wanted to create in the bay. And then also have a reproductive cells. And I think that's probably more important because we're not going to be releasing clams for the rest of our life. If we have a source of places where the babies will come out, that's what really we'll be accomplishing this. This is the first phase of our restoration program. We started in 2017 buying clams and large clams, and we were releasing 227,000 clams. That was in 2017, and then in 2019 we did 93,000. So we were moving clams from the source from Pine Island, where we were growing clams, to the Sarasota Bay. The second round, with the help of the Forensic Foundation, they provided us some fundings to buy 1 million clam babies. We bought it, the babies, put it in Pine Islands, the clam farmer raised them for us, and then in 2020, we harvest them 200, and the bottom line is that we harvest 265,000 clams that we move all over to Sarasota Bay, from Pine Island to Sarasota Bay. Now, there's a whole conglomerate of pictures here that the sites where we were putting them are the same sites that the new college student, Kara Pascarelli, with the help of Sandra Gilchrist from New College, they did a study to find the suitability site. And those are the 4 points that you see here. Those are study sites where we put clams and we see if the clams survive, grow, and they're taking pictures. And we release those clams, and you see a bunch of people here helping us. The big Budweiser truck came with the clams in the air conditioning system, and we put it displays those clams, put the clams or release the clams in these squares right on the sites that we have. So we have a specific 50 by 50 meter area where we were condensing those clams. And this is a very busy, but I wanted to just present the expenses of doing this movement. All these numbers that you see here, they're all donations by the clam farmer, by the refrigerator truck, by volunteers, divers, everybody, even a barge. All that costs money. The bottom line is $97,000 would cost the Sarasota Baywatch donations, in-kind donations. So just to let you know how much that costs, just the movement of the clams. So for phase 2, we are purchasing the clams. We're planning now, instead of just taking clams from Pine Island in there, we planning to purchase the seed clams directly from the hatchery, rear them in at least for 2 years in a predator-resistant shell length to about the size of 60 millimeters, which is predatory resistant so far. And then do it in a lease site by the Department of Agriculture and Consumer Service for restoration in Sarasota Bay. And that's their next phase that we have, is to put these clams in a location in Sarasota to let them grow, and then apply for the special activity license where we can take those clams and release them in locations where we want to put them. That concludes my presentation. In fact, I want to thank everybody, but also I want to tell you that we are collaborating with other researchers, because since we have clams, and that's where the next part is going to be Matt Ajamian, who's going to be giving us a presentation that he utilizing our clams locations to do more research on the predations of the clams. And I just want to put and stop the sharing here. If you want me, I can send you this PowerPoint so people can take a look a little more details, the things that I just.
Wonderful. No, yeah, Ernesto, thank you so much for sharing that with us. We definitely, I think we're more than happy to collect all all the visuals or information shared in the group, and definitely have it accessible in a folder where everybody can access it. So we'll start placing those today. So yeah, feel free to go ahead and send it to us, and we'll go ahead and we share it there. Thank you for telling us about your project. And now I want to invite, if anybody has a question, I haven't seen, let me see. I think some of the questions might have been for the previous speaker. But perhaps now that you've been back there afterwards, have you seen any seagrass encroaching this location at all, or is it maybe too early to say? I mean, I know this just happened at the end of last year.
Correct. We just starting to do a monitoring on the site that we released the clams. So now we are having transects and then determining how much is the survival from that first batch. And we're getting some numbers. It's kind of disappointing to see the clams being killed by predators, but there are native animals who live in that area, and we might be feeding them. What we're hoping is to increase the numbers so they can survive the predation. But that's what we're doing now. We're monitoring the growth and also the survival for those clams that we just released.
Wonderful. I have a question from David Hurls. Ernesto, I know some of the bushels you have received were smaller clams than anticipated. How do you, will you monitor or protect against predation? And I guess that you can answer that, and that will probably lead us really well into the next little presentation.
Correct. Well, quickly, so we released these clams without no protection, and the clams were about 2 inches to about an inch and a half to 2 inches. So that was the size that we have available, and that's the size that we was hoping that to be predator resistant, and not necessarily, because some of them have been dying. So yes, there were some small ones, and it happens to be there. I mean, all what we're doing is whatever is available, we're moving them out. So our next plan on the second phase of this project is to protect them more and to maybe keep a better watch for the success of these guys surviving and reproducing.
Yeah, I know that. I guess that's part of your second phase of the next round, where you're gonna intentionally like grow the clams directly in Sarasota Bay Watch and protect them until they are a bigger size, and hopefully they can, to some extent, I guess, escape some of the predation happening. So we'll see how that compares to the initial findings. I think it's going to be really interesting to see coming up.
Great.
Well, thank you so much, Ernesto. And yeah, if somebody else asks a question or anything like that, I'll let you know, and so that you can jump in and answer that. But I think this will be a nice transition to invite Matt Ahemian or Ajemian. Again, sorry for not pronouncing your last name great. He's from FAU, and he's been working also in collaboration with Sarasota Bay Watch from the post-monitoring aspect of predation. So Matt, take it away.
Great, thanks. You guys can hear me okay?
We can.
Well, yeah, my
presentation.
Okay, great. My pleasure to present this work to you guys, and really Ernesto and my colleague Kim Bassos-Holt from Moat Marine Lab and Angela Collins and Dinorah are all involved with this effort in some degree. And I was pleased to see some folks, chiming in during the session after Dr. Arnold's amazing talk and kind of synthesis of what's going on, but thinking about the predators and how they're kind of interacting with all this, which has been, it's been a fleeting thing because it's so challenging to do and takes so much effort. So I'm going to tell you a little bit about, just briefly, some work we're doing that was funded by Florida Sea Grant to have a look at what these animals might be doing at these sites. And obviously, when I'm talking about predators, because we've talked about every size of bivalve or life stage of bivalve at this point, we're talking about after they get bigger and successfully recruit to the benthos. So as they grow, I mean, there's quite a few predators, and rays are something that I've focused on in my career. I did my dissertation work on cownose and eagle rays and how they interact with shellfish in various parts of the Gulf of Mexico as well as Bermuda. But so they're going to be a focus, but obviously there's many other species of fishes, even turtles, and obviously a wide variety of invertebrates that prey on these animals. as they get bigger, of course, there's fewer and fewer of these animals that have the capacity to break open these shells. Obviously, some drills and things like that can still affect even really large clams, but we're going to focus on the rays for right now. And I've spent my career trying to figure out how to study these interactions because they are so hard to do. And obviously I've done, or not obviously, but I've done anything from predator diet analysis to tracking their movements around clam beds and things like that using telemetry systems, manipulative experiments where we exclude these predators from clam beds or other bivalve beds or manipulated beds and look at, what sort of effects they're having. And, I've even looked at, shell breakage analysis, and these have all been, they've all had their drawbacks, let's say. And, obviously when you're trying to look at predators and their impacts on these activities, you generally have to sacrifice them. And then obviously what you're looking at in their stomach contents is just their last meal, which can be really problematic for things that move, several kilometers in a day, like some of these fishes. And then even when you're tracking their movements over, these types of habitats, just because they're there doesn't necessarily mean they're eating those things. So that can be a challenge. And then these more habitat-based perspectives of how these animals are, disrupting these activities can be really field intensive. And, really trying to figure out what the predator is can be a challenge too, because, you can't be underwater watching them all the time. But, there's been some developments with passive acoustics, so listening underwater, and I'm going to talk just a little bit about how that's kind of folding into this Sarasota predator monitoring project. And we're really going to focus on the eagle ray, which is a species we've been working on, and we're hoping to expand to other predators as well. But this is a species that does eat mollusks, and in fact, about 90% of their diet is dominated by them. This is some preliminary data that one of my students is collecting or has collected along with Kim Bassos-Hull from Mote Marine Lab. And they do eat hard clams, so it's difficult for us to identify this, but we can use genetics to look at what's going on, and we have identified hard clams in their diet, but also a host of other bivalve species and gastropods as well, which is very interesting. And then here's a clip of them eating clams in captivity. And you can kind of get a sense of what this sounds like. And obviously it starts with kind of this loud pop, right? And then followed by this kind of like grinding noise. And we actually quantified this and recently published this work in the Journal of Experimental Marine Biology and Ecology. So what we're hoping to do though is now we have built this sort of library of sounds, we're trying to see if we can use passive acoustics as a predator monitoring tool in concert with other activities. So that's really important to say is, Ernesto's group is going out and monitoring, the actual densities of clams that are out there, and also looking at traces of predation. But we're hoping that these unique tool sets are going to be able to allow us to evaluate, whether predators are attracted to these sites and their potential effects on them, because that's really controversial. Not really many people are able to isolate this, but with other species of rays, as people have pointed out, like cownose, there's been some significant disruptions to restoration activities, particularly in the Chesapeake Bay area. So this is the same map that Ernesto showed earlier. These are the 4 restoration sites through Sarasota Bay. These circles here, these are showing you activity of spotted eagle rays in particular. And you can see it gets pretty high at the inlets. So this kind of set the stage for a decent experiment where we could look at how their effects are potentially changing over this sort of inshore to offshore landscape. So we deployed a bunch of these passive acoustic recorders, and this one here is called the Cyclops. It actually has a camera on the other end of it and the hydrophone obviously on this left end. And we did this, we tried to really time this really well. So it took significant coordination with Sarasota Baywatch. So I want to really thank them again. And we were able to get out and deploy these systems before and after these clam releases, which took place sort of at different intervals across 3 sites in particular. We also had an opportunity to deploy this gear at these reference sites about 1 kilometer away, where we knew we wouldn't be hearing activity on the restoration site. So we had a couple of those reference sites in there. And you know, we're just getting into the data, but this here is called a spectrogram. So you're looking at the frequency on the y-axis and then time on the x-axis. And what you're seeing here is an event that I isolated right after the restoration occurred on September 26th. So I'm just going to let you hear that. So you're hearing a pop and then some sort of grinding and crunching there. And just to give you some perspective, that's from the field. And this is sort of what you hear in captivity. Pop is much closer and louder, and some more grinding. So we think that there's some evidence that, you know, the animals are feeding in this area. Certainly they can be feeding on other things because we found, you know, other bivalves there as we're going around and collecting. But the key is that the acoustics match in the field what we've recorded in captivity, and you know, certainly matches what we would hear for hard clams, which we fed these animals. So, you know, somewhat promising, but a lot more work to do. And then these are the presence of different rays. Each individual here is a different color at these restoration sites. So site 4, and you see these blue arrows, these are events when clams were released. So, you know, clearly these animals are still using these areas as these events are going on. So we're going to take a look at that, as well as we've developed a detector. So these acoustic systems, they record data for, you know, days on end, and the data is quite intensive and requires you to have special tools, in particular computer algorithms, to really sift through and find these events because it is so much data. So we're going to apply a detector we developed to that so we can better identify events. So I'm not just sitting there listening to every single recording. And then we want to look at how these consumption rates may change over time and space. We hypothesize that, again, as you move away from these inlets, which are kind of these access ways for these predators, that, you know, these rates will potentially go down. And then we, again, have these reference sites. So really comparing consumption rates between, you know, the restoration and reference sites is a major goal. And then we're obviously going to continue coordinating with Sarasota Baywatch to look at how these sort of activities may relate to what they're seeing in terms of evidence on the ground. One of the challenges with some of these larger predators is sometimes they'll grab these clams and swim away with them and crack them. Obviously, they do that usually within a couple of minutes, but, you know, it can be a challenge by using the fragments alone to determine what they're doing. Also, sometimes those shells are pulverized into really small pieces. So this is where the acoustics comes in as a potential tool to track some of that. And then we're hoping to submit this work to a much larger funding opportunity using this pilot data. So I just want to thank Dinorah and Angela. You guys have been really good, really supportive. Thank you so much for that. I guess in getting me into the program here, and all of these individuals and support mechanisms that are funding the work. And if you guys are interested more in what we do, you can check out our website. We're also on, you know, Instagram, or feel free to send me an email if you're interested in any of this work or applying it to other systems or situations. So thank you.
Wonderful, thank you, Matt. That was excellent to see, and so exciting, the results. That looks really promising, and also that you can have that before, after, or comparing into other sites as well, and seeing how that may change or be similar, or, you know, like really letting us know what happens after we put all this effort of putting all these clams out there. Are we just feeding all the race populations? What is really happening?
Yeah.
So I think, what's that?
Thank you very much.
Absolutely. Thank you for taking the time and letting us know how these projects go hand in hand. I think it's definitely insightful, and things that we also need to think about in terms of different, like the trophics and, you know, trophic structure and what is happening with with those animals that actually do feed on the clams, and all of our efforts of restoring clams as well. Alrighty, if I haven't seen any other questions, but there, Angela just posted Matt's information. I will also populate a list of all the participants and their emails, and so that everybody can have access to each other's. But feel free to go ahead and keep posting on the chat box your information or anything that you think might be relevant to the group. Now we're gonna switch coasts, and we're going to the East Florida in the Indian River Lagoon. I hope that we have Todd Osborne from UF in the audience. Todd, if you're around, feel free to jump in and let us know what you guys are doing on the other side of Florida.
Hey, how are you? Well, thanks for giving me a chance to tell us a little bit about tell you guys a little bit about what we're doing here. I'm here this morning with Mike Sullivan from Premium Seafood, who's our industry partner. So there are farmers and industry folks present today. But our group is a pretty big association. So we've got friends with NEP and FWC and the Water Management District, as well as Fish America and CCA and a bunch of different groups that are working together to try to bring clams back into the picture in the IRL. And we've been hammering away at this slow but sure since 2016. So there's been a lot of prior planning that kind of went into the last 2 years of funding that we've had to do work in the IRL, and thus far things have been pretty successful. Based on we've already heard from Dr. Arnold, and that one of the main strategies, and I'm glad that he said this because it influenced us a lot, was to get reproductive islands going back in the system, and by that meaning a high density population restoration in areas that have been not the hardest hit in the IRL system, but coming in from the ends, meaning down from the Edgewater area, heading south through Mosquito, and then up from Sebastian, where water quality is a little better. And we're trying to move in and bring these kind of high density areas where we replant native clams and get these populations kind of bolstered back up. And as Bill said, it doesn't take a lot to get a lot of recruitment, but the idea is that we kind of keep moving toward the areas where we know water quality was the worst. And I guess the strategy is probably what's unique about this particular project in our direction, and that was to utilize a very rare, what we thought was very rare find of a couple of broodstock in southern Mosquito Lagoon that had been subjected to some tremendous environmental insults over the last years. I mean, we're all familiar with the super blooms in 2011 and 12 that really just wrecked the Indian River Lagoon. And after that, finding a live clam was something of a unicorn chase. And I think our staff put in about 2 months of time, many hundred man hours to come up with 39 large adult mercenary clams that we use as broodstock. And so between my biologist, Jose Nunez, who's an aquaculture specialist, and Mike Sullivan at Premium Seafood, we were able to spawn those 39, and we have put 4 million of those back in the IRL to date. And actually, it was surprising, even though we had this big algal bloom this past year, things went really well, with the exception of one of our sites that's kind of central, close to Melbourne, that experienced a really dramatic change in salinity. So, of course, 18 inches of additional rain this year fell in the Brevard County area, and that dropped our salinity down to 12 for a while around one of our major sites, and it took a pretty tremendous shot there. But all in all, I think we're still running around 75 to 80% survival across the board, which is great. And I think that we're going to start villager assessment this year, but we're looking to see if anything is coming out of those leases now. Now, one big problem that I think has been touched on already today, and I hope we have time to discuss more in this process, is that there is a permitting issues between putting out clams in different areas. So our first batch going through aquaculture, of course, has been used and distributed to partners that are working with us that have actual FDACS approved clam leases. aquaculture leases. And so we started there. Now, since then, I guess last year, we're able to find some wild caught broodstock that we can confirm are wild caught, and then we can work with FWC to place those in a rather large project that we have going on with Brevard Zoo in much more diverse, spatially diverse areas. But that has been a bit of a hurdle to work with. And I want to say that FDAX has been really helpful working with us on that. And they've come to the table, Sea Grant's come to the table to help with that as well. And we even did some transplanting of clams from farmers around the state as part of a COVID economic relief through Sea Grant to put clams back in the Titusville area this past fall. So there's been a great interest from the public. We do have Blair Wiggins working with us, and he's been a huge supporter of this project, and he has a addictive fishing television show that has a lot of followers. So the public's been pretty vocal about, hey, we like this idea, we want to support it. And I can't say enough about all the agencies that have poured in time and effort to get behind this. And I think we all appreciate what Dr. Arnold was saying about, there's a strategy to this. It's not just dump clams in the water, but we really have to work on the drivers that are keeping them out and try to identify those, understand those, but building that kind of understanding into our strategy of repatriation is really important. And I think that that's kind of what we're trying to do in the IRL. And I think that I feel like in this group today, just from what we've heard already, we're all on the same page and why we want to do this. But there are some significant hurdles to overcome scientifically as well as policy-wise, and I think those are getting touched on some more talk. So anyway, if anybody wants to reach out and talk more about what we're doing in the IRL, please hit me up with an email or give me a call, and be glad to elaborate more.
Wonderful, Todd. That was a great overview of you guys' project and effort. I wanted in this moment, I guess, to reach out to if we have anybody from FDAX or FDEP or anybody who would like to comment a little bit on the permitting aspect, and if anything that you might want to add to that on that regard. I know that we will be hearing a bit more about from the scientific aspect of this ecological services, but I think we'd definitely love to hear more about you guys from the, I guess, policy side or permitting aspect. It seems like it's also taking some time, or it's not as developed, or perhaps it's just something else that it's currently happening. So feel free, anybody if you have anything to add or comment about that, you have the floor. Okay, I see a question about what about leasing specifically? Maybe is this perhaps the difference between,
Dinorah, this is Marcy.
Okay, hi.
Hi everybody, Marcy Cockrell from the FDAX Division of Aquaculture. I just meant, what specifically about leasing are you looking for comments on from FDAX? I just wanted some clarification, and I think we might have other folks from aquaculture too on the webinar that are more versed in this than I am. I just wanted to get some clarification about what you were looking for, what kind of information.
Okay, I think it might be perhaps, and Todd, you let me know if I'm talking about the right thing, but perhaps like differences between the leases. I know there's been perhaps some confusion between what's considered aquaculture lease versus sort of the restoration aspect or that sort of thing. Is that some of a bit of the hurdles, or what do you think, Todd?
I think, well, I don't view it so much as a hurdle as we had to learn the ropes, so that if you're bringing products that have an aquaculture lineage out into the environment, then we were under the understanding, of course, that we need to work through FDAX and utilize our partner leases as a place to develop those populations. Now, wild caught broodstock would have a different angle on permitting for release, and so that would fall under FWC. At least that's my understanding to date. And so we've come in working parallel both with FDAX and utilizing for conservation purposes or restoration purposes some administratively closed lease areas, like in the Titusville area for the Sea Grant project. And that works really well because there's some added benefit to being on a lease that has some protection to it and things like that. And most of the public's aware they can't go out and just willy-nilly take clams off of leases. But in the same vein that when we were working with wild caughts that don't have any aquaculture lineage, then that falls under the purview of permitting for for FWC. And so we're kind of working through that second part now. We have a big project with the Brevard Zoo and FOS, and where we're doing seagrasses and clams together. And those are done on small parcels, or planning to do on small parcels with individual clam gardeners versus the oyster gardeners that they've been working with on various other restoration projects. And so individuals monitoring their own little patch, their own little population on their own shoreline. So that was working through FWC versus working through FDACS for the aquaculture side of things. And same thing, there's just those not necessarily hurdles, but challenges that we had to understand the process. And that was kind of a thing that took us a while to get to. But like I said, both agencies have been working with us significantly, and we're really happy to have those associations. I also want to mention one thing if I can, because some of my buddies from USDA are on the call today, and there's a product or a project going on that's going to produce a product that I think will be very valuable to everybody involved here. The NRCS group, or Natural Resource Conservation Service, is doing a subaqueous soil survey. That's a mouthful, but what that means is they're looking at soil attributes all around, submerged lands around the state of Florida, and specifically right now, IRL area is a focal point, but there's also a group based in Southwest Florida that I believe is going to be working in that area as well. I think Martin is on the call today. He can speak to that. But one of the products that comes out of that is this subaqueous soil survey is looking at areas that are suitable for restoration of seagrasses and shellfish, as well as aquaculture leases, if that would be something that come out in the future. So that might be something that helps work in synergy with the folks that are doing the suitability modeling, et cetera. So there's a whole lot of different pieces that I think as a group we can pull together to make great advances here.
Absolutely. I think that's a wonderful thought. Thank you for all the information. And yeah, and Marcy, she just reached out and said that it's a great summary. If anybody has any questions about the process of permitting or any of that, feel free to reach out to FDACS, and they're happy to work with people through the process. I know that that's a topic that has come up recently a lot, depending on exactly what you're doing in the way that you're restoring, it definitely can change the path or who you're working with and what applications and under what regulations. So it can definitely be a bit tricky, but I as Marcia and Todd said, FDACS and FDEP and all, they're all very helpful and happy to work with all you guys. It's just like a little bit of tweaking on how to streamline the process, I think, to make it easier and not so, I guess, bumpy to figure out how to do what in what way. So it all depends on what you're doing and how you're doing it. So I think that's a trick right there. They're asking for your email, Todd, if you can add it to the chat box. Again, I will make sure that we distribute a list of all the participants and their information. We have gotten some other documents shared between studies and fact sheets and great funding information for, I guess, possible sources of funding, also shared in the chat. So if not now, I will make sure that we have all this in the folder, and if not, at the end, I will make sure that we have all that in a document so that you guys can have access to all that great info down there. But I think we're reaching out to the end of our morning session. I want to thank absolutely everyone. Before we close, I just want to reach out in the audience if there's any other groups that I might have missed. And first, I apologize for that. If somebody...
Hello, Dinorah.
Hi, Curtis.
Hi. Yeah, I just wanted to make just share something specifically with Todd and the group.
Yep.
Let you just to give Todd the update on what's been going on in the West Coast and how we've been dealing with permitting for the release of bivalves over the years. Certainly, we used FWC primarily when we were doing bay scallop restoration over many, many years. But with bivalves, and you can also reach out to Ernesto over at Sarasota Baywatch, since they've been doing the same similar work to yours on the East Coast over the same time period. But what we've been doing is we approached it at FDACS, and Charlie Culpepper's been spearheading being able to create restoration leases for a product that's aquaculture. It doesn't have to be wild. You can aquaculture product and put it down, and they're just now, they're halfway through it. I believe they just received or were able to make some language change, or they're about to get it approved through the Board of Trustees. I'm not sure the exact stage, but I know it's one step forward for sure. And then outside of that, the other release areas that would not be in, primary aquaculture leases, DEP has allowed us on the West Coast to be able to put bivalves into the environment in approved or non-shellfish approved waters through their general permit, and they're actively looking at creating a more specific method for applying to do this type of work under the general permit, so it reduces the amount of time and the understanding of what we're asking to be permitted. So I hope that helps. And again, Ernesto can give you the person's name at DEP too, that's been our lead contact on that. Thank you.
Perfect. Thank you so much, Kurt. And those for you in the audience who don't know Kurt, he owns Bay Shellfish. He has a hatchery down in Terracea. So he's also been involved in a lot of the supplies for clams and bivalves over the different restoration projects, a lot of them going on the West Coast. So pretty well connected to all of our partners. And yep, now Angela just shared their website. And I want to, say thank you to everyone who attended this morning. I hope that this has been as instructive for me as has been for you guys. And we love to continue the conversation in the next 2 days. It will be a little bit of a different structure, which I will go over that early first thing tomorrow. But if nobody else has any other last quick remarks for our first day, I want to thank you guys for being present. Also, I want to thank Arnold Bale for all his knowledge that he shared and time with us today and his advice. I also want to thank all the different representatives for the restoration groups here that were able to give us overview of what is happening all around Florida. I think that we can agree that we have more in common than we think. There's a massive effort and movement on clam restoration, and there's still much that we need to figure out and to move forward in this field. So hopefully with this establishes a really good connection among all of us, and please feel free to reach out to us or to each other. So this is the main goal of this project, of this workshop. And without further ado, thank you guys for attending, and I will see you tomorrow bright and early at 9 a.m. if you can make it. I will hang out a little bit longer if anybody has any comments, but feel free to go ahead and leave as you please. Nice. Thank you, everyone, for your comments. Take care, guys. Thank you.